The pressure at the bottom of the Mariana Trench exceeds 1,000 times the atmospheric pressure at sea level. Let the weight of the day drift away, and let your body settle into the dark. We are going somewhere. There is time. The lights can stay dim. The ocean is a realm of layered darkness, a vast and silent universe that covers more than 70% of our planet's surface. Yet we have mapped the surface of Mars with greater precision than we have mapped the seafloor of our own world. There is a region within this watery world where the conditions are so extreme, so unforgiving, that scientists once believed no living thing could possibly exist there. They called it the Hadal Zone, named after Hades, the ancient Greek underworld, the realm of the dead. And for decades, this name seemed fitting, for the Hadal Zone is the deepest, darkest, and most hostile environment on Earth. The Hadal Zone begins at a depth of 6,000 meters below the surface of the sea. This is not a depth that occurs just anywhere in the ocean. It is found exclusively inside the great ocean trenches, the deep scars that run along the boundaries of Earth's tectonic plates. These trenches are the deepest places on the planet, and they are remarkably rare. The Hadal Zone constitutes less than 2% of the total ocean floor, yet it accounts for over 40% of the ocean's total depth range. This threshold of 6,000 meters matters physically and biologically in ways that are profound. Below this depth, the pressure exceeds 600 atmospheres, a force that would crush almost anything we know from the surface world. The temperature hovers near freezing, and the darkness is absolute, eternal, and complete. There is no light, no warmth, no photosynthetic life, and almost no food. And yet, as we have come to discover, life persists here in ways that challenge our understanding of biology itself. Let us begin with the geography of these trenches, for they are the gateways to the Hadal realm. The most famous of them is the Mariana Trench, located in the western Pacific Ocean, south of Japan and east of the Philippine Sea. It is the deepest known point on Earth, reaching a measured depth of 10,916 meters at a location called Challenger Deep. To put this in perspective, if you were to place Mount Everest, the tallest mountain on Earth, at 8,848 meters into the Mariana Trench, its summit would still be more than 2,000 meters beneath the surface of the waves. The trench itself stretches for approximately 2,500 kilometers, a crescent-shaped scar in the ocean floor that runs from Guam to the northern Mariana Islands. The Tonga-Kermadec system lies to the east of New Zealand in the southwestern Pacific Ocean. This system is composed of two connected trenches, the Tonga Trench and the Kermadec Trench, which together form one of the longest and deepest trench systems on Earth. The Tonga Trench reaches a maximum depth of 10,882 meters, making it the second deepest point in the ocean. The system stretches for more than 2,500 kilometers along the boundary where the Pacific Plate dives beneath the Tonga Plate. This is also one of the most geologically active regions on the planet, with frequent earthquakes and volcanic activity driven by the relentless subduction below. The Philippine Trench, also known as the Mindanao Trench, lies in the western Pacific, east of the Philippine Islands. It reaches a maximum depth of 10,540 meters, and it runs for approximately 1,300 kilometers in a north-south direction. This trench marks the boundary where the Philippine Sea Plate is being forced beneath the Sunda Plate. The trench is relatively narrow, but its walls are steep and dramatic, descending from shallow continental shelves to abyssal depths in a very short horizontal distance. The Kuril-Kamchatka Trench runs along the Pacific coast of Russia, from the Kamchatka Peninsula southward to the northern islands of Japan. It reaches a maximum depth of 10,542 meters, and it extends for approximately 2,200 kilometers. This trench is formed by the subduction of the Pacific Plate beneath the Okhotsk Plate, and it is surrounded by a chain of volcanic islands that are among the most active on Earth. The trench is notable for its steep slopes and its position within a region of intense seismic activity, including the great earthquakes that have historically struck the Kamchatka Peninsula and the Kuril Islands. Finally, the Puerto Rico Trench is the deepest point in the Atlantic Ocean, reaching a maximum depth of 8,376 meters. It lies at the boundary between the Caribbean Sea and the Atlantic Ocean, north of Puerto Rico and the Dominican Republic. Unlike the Great Pacific Trenches, which are formed by the subduction of one oceanic plate beneath another, the Puerto Rico Trench is a more complex geological feature, involving subduction of the North American Plate beneath the Caribbean Plate, but also elements of transform faulting. This trench is the deepest single point in the entire Atlantic, and it is the only one of the 5 major Hadal trenches that lies outside the Pacific Ocean. All of these trenches, and the many smaller ones that also reach Hadal depths, are formed by a process called subduction. This is one of the most fundamental and powerful forces in geology. The surface of the Earth is composed of a mosaic of tectonic plates, massive slabs of rock that float on the semi-molten mantle beneath. These plates are in constant motion, drifting at rates that are measured in centimeters per year, about the same speed at which your fingernails grow. Where 2 plates collide, one will often be forced beneath the other, diving down into the mantle in a process called subduction. The descending plate is the heavier, denser one, typically an oceanic plate made of dense basaltic rock, while the overriding plate might be composed of lighter continental rock or another oceanic plate. As the descending plate plunges into the mantle, it drags the sea floor with it, creating a deep depression in the ocean floor at the boundary between the 2 plates. This is the trench. The process is slow, relentless, and continuous, taking place over millions of years. The trenches we see today are the result of subduction zones that have been active for tens of millions of years, and they continue to evolve even as we speak. Now let us consider the conditions that make this realm so inhospitable. The first and most immediate threat is pressure. At the surface of the ocean, we experience the pressure of Earth's atmosphere, which is about 101,325 pascals, or approximately 1 atmosphere. At the bottom of the Hadal zone, the pressure increases by roughly 1 atmosphere for every 10 meters of depth. At 6,000 meters, the pressure is about 600 atmospheres. At the bottom of the Mariana Trench, at 10,916 meters, the pressure exceeds 1,100 atmospheres, or about 110 megapascals. This is the equivalent of having about 1,100 kilograms of weight pressing down on every single square centimeter of surface area. To understand what this means physically, consider the deepest pressure we can experience on land. The pressure at the bottom of the deepest mine on Earth, the Imponing gold mine in South Africa, is about 4 kilometers deep, and the pressure there is less than 1/10 of the pressure at Challenger Deep. At Hadal depths, the pressure is so intense that it would crush a human being in an instant, flattening the lungs, collapsing the rib cage, and destroying every air-filled cavity in the body. It is a force that our bodies are entirely unprepared to withstand, and yet the pressure is not the only hostile condition. The temperature in the Hadal zone is near freezing, typically between 1 and 4 degrees Celsius. This temperature is remarkably stable, remaining constant throughout the year because the deep ocean is insulated from the seasonal variations that affect the surface waters. The water at Hadal depths is a mixture of cold, dense water that has sunk from the polar regions, where it chilled at the surface and then descended, and deep water that has been slowly circulated by the global ocean currents. There is no source of heat in the deep trenches, aside from the occasional volcanic vent, and so the temperature remains locked in this narrow, frigid range. For any organism that lives there, the cold is a constant challenge, slowing metabolic processes and making survival a matter of extreme efficiency. The darkness of the Hadal zone is absolute. Light from the sun penetrates the ocean surface and is gradually absorbed by the water. By a depth of 100 meters, most of the light has been absorbed, and by 1,000 meters, the ocean is completely dark, with no trace of sunlight remaining. The Hadal zone, at 6,000 meters and below, exists in a state of perpetual, eternal night. No plant can photosynthesize in this darkness, and so there is no primary production of food through photosynthesis. The only sources of light in the deep ocean are bioluminescence, the faint glow produced by certain organisms, and the occasional flash of a hydrothermal vent. But these are rare and localized, and they do not provide enough energy to support the kind of food webs that exist in the sunlit surface waters. The absence of light means the absence of photosynthesis, and the absence of photosynthesis means the absence of the base of the food web that sustains life in the ocean's upper layers. The Hadal zone is therefore almost entirely dependent on a slow, trickling rain of organic matter from above. This is called marine snow, and it is the only substantial source of food for the deep-sea creatures that call the trenches home. Marine snow is composed of tiny particles of organic material, including dead plankton, fecal pellets, fragments of shells, and other detritus that falls from the surface waters. It descends slowly, at a rate of perhaps 100 to 1,000 meters per day, and it takes months, sometimes years, for this material to fall the full 11 kilometers from the surface to the bottom of the deepest trenches. During this long descent, the marine snow is consumed by bacteria, by scavengers, by filter feeders, and by any other organism that can find it. The caloric value of the snow is almost entirely depleted by the time it reaches the Hadal zone. What arrives at the bottom is a pale, nutrient-poor sediment, barely capable of supporting life at all. And yet, life does exist there, in forms that are still being discovered and understood. Our understanding of the Hadal zone has been built over more than a century of exploration, and the history of this exploration is as remarkable as the environment itself. The first systematic attempt to measure the depths of the ocean came with the HMS Challenger expedition, which set sail from Portsmouth, England, in December of 1872, under the command of Captain George Nares. The expedition was a scientific endeavor of unprecedented scale, funded by the Royal Society and the British government, with the goal of exploring the physical, chemical, and biological conditions of the deep sea. Over the course of the next 4 years, the Challenger traveled more than 127,600 kilometers across the world's oceans, from the Atlantic to the Pacific, from the Southern Ocean to the tropics. The crew conducted 492 individual deep soundings, using the most advanced technology available at the time. This meant lowering a heavy weight, usually a cannonball, attached to a length of hemp rope over the side of the ship, and measuring how much rope was needed to reach the bottom. The process was slow and painstaking, requiring hours of labor for each measurement, and the results were often imprecise. Hemp rope stretches under tension, and the ship could drift during the measurement, making the readings unreliable. Nevertheless, the Challenger expedition made history. In 1875, the ship conducted a sounding near the Mariana Islands that recorded a depth of approximately 8,184 meters, which was the deepest measurement ever taken at the time. This point was later named Challenger Deep in honor of the expedition, and it was eventually recognized as the deepest point in the world's oceans. The methods of the Challenger expedition were primitive by modern standards, but they established the foundation of deep-sea exploration. In the decades that followed, the hemp ropes were gradually replaced by steel wire, which was stronger, less stretchable, and more reliable. Steel wire allowed for more accurate measurements and deeper soundings, and it became the standard tool for oceanographic surveys in the late 19th century. However, even steel wire had its limitations. Lowering a weight to the bottom of the ocean and hauling it back up was a slow process, and it could only provide a single point of measurement at a time. Mapping the seafloor in any meaningful sense was impossible with this method. The next revolution in depth measurement came in the early 20th century with the development of echo sounding. In 1914, the Canadian-American inventor Reginald Fessenden demonstrated the first practical echo sounder, which worked by sending a pulse of sound down into the water and measuring the time it took for the echo to return from the seafloor. The speed of sound in seawater is known, approximately 1,500 meters per second, and so the depth could be calculated from the time delay. Fessenden's device was initially developed for military purposes, specifically for detecting icebergs and submarines, and it was used during the First World War for this purpose. But after the war, the technology was adapted for oceanographic use, and by the 1920s, commercial echo sounders were being deployed on research vessels, allowing scientists to map the seafloor continuously as they traveled. This was a transformative breakthrough. Instead of taking a single-point measurement every few hours, a ship could now record the depth continuously, creating a detailed profile of the ocean floor along its entire track. The era of modern bathymetry had begun. It was with this technology that scientists began to discover the true scale of the Hadal trenches. In the 1920s and 1930s, surveys of the Pacific Ocean revealed the extent of the Mariana Trench, the Tonga Trench, and the other great deeps, and the depths they recorded were staggering. The deepest point of the Mariana Trench was measured at over 10,000 meters, a figure that was almost beyond comprehension at the time. The term Hadal Zone was coined by the Swedish oceanographer Hans Pettersson in the 1950s, and it entered the scientific literature as the name for the deepest and most extreme habitat on Earth. But knowing the depth of the trenches was only the beginning. The first human visit to the Hadal Zone came in 1960, and it remains one of the most remarkable achievements in the history of exploration. The vessel was the bathyscaphe Trieste, a deep-sea submersible designed by the Swiss physicist Auguste Piccard. Piccard had built his first bathyscaphe in the 1940s, and the Trieste was the culmination of his work. It consisted of a large, cigar-shaped float filled with gasoline, which provided buoyancy, and a small spherical pressure sphere attached to the underside, which carried the crew. The pressure sphere was made of thick steel, designed to withstand the immense pressures of the deep ocean. The Trieste was purchased by the United States Navy in 1958, and it was modified and strengthened for operation at extreme depths. The crew for the historic dive was Jacques Piccard, the son of Auguste Piccard, and Lieutenant Don Walsh of the United States Navy. On January 23, 1960, the Trieste was positioned over the Mariana Trench, near the island of Guam. At 8:23 in the morning, the descent began. The bathyscaphe sank slowly, taking on water ballast to increase its weight, and it fell through the water column at a rate of about 1 meter per second. The descent took nearly 5 hours. As the Trieste sank, the crew watched the depth gauge, and the temperature outside the sphere dropped to near freezing. The light faded, and the darkness became absolute. At around 9,000 meters, the crew heard a loud cracking sound, which they later determined was caused by the plexiglass window in the entrance hatch cracking under the pressure of the water. The window held, but the moment was tense, and the crew considered aborting the dive. They decided to continue. At 106 in the afternoon, the Trieste reached the bottom of the Challenger Deep, at a depth of 10,916 meters. The pressure at that depth was approximately 1,100 atmospheres, a force that would crush any ordinary vessel. The Trieste, however, had been designed for this moment. The crew spent 20 minutes on the bottom, observing the seafloor through the small porthole. They saw a pale, silty sediment composed of diatomaceous ooze, the remains of countless microscopic algae that had rained down from the surface over millions of years. Jacques Piccard later reported seeing a flatfish, a type of bottom-dwelling fish, lying on the seafloor. This observation was met with great excitement, as it suggested that even the deepest point of the ocean could support complex life. However, the observation was later questioned by scientists who pointed out that the visibility from the bathyscaphe was limited, and that the creature Piccard saw might have been a sea cucumber, a holothurian, which is a type of echinoderm rather than a fish. The debate continues to this day, but the dive itself remains an undisputed milestone in human exploration. After 20 minutes on the bottom, the Trieste began its ascent. The crew released the iron ballast, which allowed the bathyscaphe to rise, and the ascent took approximately 3 hours and 15 minutes. The bathyscaphe surfaced at 4:56 in the afternoon, and the crew emerged, having made history. They had become the first and only humans to reach the deepest point of the ocean, and they had done so more than 50 years before anyone would repeat the feat. The wait for the next human visit to Challenger Deep was 52 years. In March of 2012, the Canadian filmmaker and explorer James Cameron made a solo descent to the bottom of the Mariana Trench in his submersible, the Deep Sea Challenger. Cameron's submersible was a radically different design from the Trieste, with a single-person pressure sphere, a vertical orientation, and a suite of high-definition cameras and lighting systems. Cameron reached the bottom at a depth of approximately 10,980 meters, and he spent several hours on the seafloor, collecting samples and filming the environment. His dive was the first human visit to the Hadal Zone in more than half a century, and it demonstrated that the technology for deep-sea exploration had advanced significantly. Since Cameron's dive, there have been additional human visits to the Hadal Zone, but they remain rare, and the majority of our knowledge of this environment comes from unmanned vehicles, remote-operated vehicles, and deep-sea landers. Now, as we consider the conditions that should make survival impossible in this realm, we must turn to the molecular physics of pressure. The force of 1,100 atmospheres is not merely a physical pressure on the hull of a submersible. It acts on every molecule, every cell, every protein in any organism that lives there. Water is nearly incompressible, and so the pressure at Hadal depths is transmitted through the water column with almost no attenuation. This means that any organism living in the Hadal zone is subject to the same pressure as the water around it, both inside and out. The pressure compresses the space between molecules, and it can force water molecules into the structures of proteins, altering their shape and function. Proteins are the molecular machines of life, and their function depends on their precise three-dimensional structure. High pressure can denature proteins, causing them to unfold and lose their function. Cell membranes, which are composed of lipid bilayers, are also vulnerable to pressure, which can alter their fluidity and permeability. At the pressures found in the Hadal zone, these effects should be catastrophic, and yet organisms not only survive but thrive in these conditions. They have evolved molecular adaptations, including the production of specific proteins called piezolites, which help stabilize proteins and membranes under high pressure. These adaptations are subtle and complex, and they represent one of the most fascinating frontiers of biological research. The fact that life can exist at all under such conditions is a profound challenge to our understanding of the limits of biology, and it raises questions that we are only beginning to explore. As we continue to study the Hadal Zone, we are discovering that the deepest places on Earth are not sterile wastelands, but thriving ecosystems, filled with creatures that have evolved to thrive in a world that should be impossible for life to survive. The molecular physics of pressure, the mechanics of protein folding, the biochemistry of cell membranes, all of these fields are being rewritten by the discoveries emerging from the deep trenches. And the more we learn, the more we realize how little we truly understand about the nature of life itself. The Hadal Zone is a realm of extremes, and it holds secrets that we are only just beginning to unveil. The most studied of these piezolites is a small molecule with a deceptively simple name, trimethylamine N-oxide, or TMAO for short. This compound is not exotic. It is, in fact, something we encounter every day, often without realizing it. It is the molecule responsible for the characteristic smell of fish, the faintly marine, slightly ammonia-like scent that we associate with the sea. When a fish dies and begins to decompose, bacteria break down TMAO into trimethylamine, and it is this breakdown product that carries the odor we recognize. But in life, TMAO serves a far more profound purpose than scent. The researcher who has done more than anyone to illuminate the role of TMAO in the deep sea is Paul Yancey, a biologist at Whitman College in Walla Walla, Washington. For decades, Yancey has been collecting deep-sea organisms and measuring the concentrations of TMAO in their tissues. His work has revealed a remarkably consistent pattern. The deeper an organism lives, the more TMAO it contains. This is not a vague correlation. It is a precise, quantitative relationship. As depth increases, TMAO concentration rises in a steady, almost linear fashion. A fish living at 1,000 meters has a certain level of TMAO. A fish living at 5,000 meters has more. A fish at 8,000 meters has more still. The relationship is so consistent that scientists can estimate the depth at which an organism lives simply by measuring the TMAO concentration in its tissues. What makes this relationship even more striking is the mathematical precision of it. Yancey and his colleagues have found that TMAO concentration increases at a rate that closely tracks the increase in hydrostatic pressure. This is not coincidental. The molecules are responding to a physical force. At depth, water pressure compresses proteins and other cellular structures, forcing them into conformations that are less stable and less functional. TMAO acts as a chemical counterweight. It is what scientists call a counteracting osmolyte. It does not directly resist the pressure. Instead, it alters the local environment around proteins in a way that makes them more resistant to compression. In essence, TMAO pushes back against the pressure-induced destabilization of proteins, restoring them to a state that is closer to their normal functional shape. The molecular mechanism is subtle. TMAO molecules are excluded from the immediate hydration shell of proteins. This exclusion means that TMAO effectively increases the free energy cost of unfolding a protein. When a protein is folded, it presents a compact, defined surface to the surrounding water. When it unfolds, it exposes a much larger, more disordered surface. TMAO makes that unfolded state energetically unfavorable. It preferentially stabilizes the folded native conformation. Under pressure, proteins tend to unfold because the compressed water molecules can interact more favorably with the exposed surfaces of the unfolded protein. TMAO counteracts this tendency. It raises the energy barrier to unfolding, making it harder for pressure to push the protein into an inactive state. This is why surface fish have almost no TMAO in their tissues. At the surface, atmospheric pressure is only about 1 bar. There is no compression to counteract. A fish that lives in shallow water does not need this chemical protection, and the cost of synthesizing TMAO is not trivial. It requires energy and resources. Evolution is economical. It does not maintain costly defenses that are not needed. So surface fish have barely detectable levels of TMAO in their muscles, and the smell of their flesh when it spoils is relatively mild. At the other extreme, the Hadal snailfish, which lives at depths of 8,000 meters, carries TMAO concentrations that are extraordinary by any measure. In some deep-sea species, TMAO can account for a significant fraction of the total organic molecules in the tissues. The concentration is so high that it would be toxic to a surface fish, but for the snailfish, it is the difference between life and death. The story of TMAO is a reminder that life at depth is not a matter of simply enduring pressure. It is a matter of active, ongoing molecular work. Every protein in every cell of a deep-sea organism must be protected, stabilized, and maintained against the constant, crushing force of the surrounding water. This is not a passive state. It is an active investment of energy, and it shapes the entire biology of the organism, from its biochemistry to its metabolism to its very existence. But TMAO is only part of the story. The proteins themselves have also evolved. This is where the work of researchers at the Scripps Institution of Oceanography in La Jolla, California, becomes central. Scripps has been at the forefront of deep-sea microbiology for decades, and one of its most distinguished researchers, Douglas Bartlett, has devoted his career to understanding how bacteria survive in the Hadal zone. Bartlett's laboratory studies piezophilic bacteria, organisms that are not merely tolerant of high pressure, but require it for growth. These are the true natives of the deep. They cannot survive at surface pressure. If you bring them to the surface, they die. Their membranes rupture, their proteins unfold, and their cellular machinery simply stops working. Bartlett and his colleagues have sequenced the genomes of these piezophiles and compared them with their shallow water relatives. The differences are revealing. The enzymes of piezophilic bacteria have amino acid compositions that differ systematically from those of surface bacteria. They have more of certain amino acids such as glycine and alanine, which are small and flexible, and fewer of the bulky, rigid amino acids that tend to lock proteins into a single, inflexible conformation. This is not a coincidence. The flexibility of these enzymes is essential. At high pressure, proteins tend to become compressed and rigid. They can no longer undergo the conformational changes that are necessary for their catalytic activity. The enzymes of piezophiles are built to resist this rigidity. They are, in effect, more flexible at the molecular level, allowing them to continue functioning even when the surrounding water is pressing in on them with enormous force. This is a fundamental insight. The enzymes of surface organisms are not simply overwhelmed by pressure. They are locked into inactive conformations. The pressure forces them into shapes that cannot bind their substrates or catalyze their reactions. The enzymes of deep-sea organisms, by contrast, have evolved to maintain their flexibility even under extreme compression. They are not more robust in the sense of being stronger or more resistant. They are more flexible, more adaptable, more capable of changing shape in response to the demands of their environment. This is a different kind of adaptation, and it has profound implications for our understanding of how life works at the limits of possibility. The distinction between piezophiles and piezotolerant organisms is crucial. Piezotolerant organisms can survive at high pressure, but do not require it. They can be brought to the surface and still function. Many deep-sea fish, including the snailfish, fall into this category. They tolerate the pressure of their environment, but they are not dependent on it. Piezophiles, by contrast, are obligate. They require pressure. Their very existence is tied to the deep. For them, the surface is a lethal environment. This is a difficult concept for us to grasp, because we live at the surface and we think of pressure as something that can be endured or resisted. But for a piezophile, pressure is not a threat. It is a necessity. It is as essential as oxygen is to us. The question of how to study these organisms, of how to reach them and observe them in their natural environment, is one of the great logistical challenges of deep-sea science. A crude submersible capable of reaching 11,000 meters, like the Deep-Sea Challenger or the Limiting Factor, is an extraordinary piece of engineering. It costs tens of millions of dollars to build and operate. It requires years of preparation, a dedicated support vessel, and a highly trained crew. The dives themselves are exhausting and dangerous. The pressure at those depths is so great that the hull of the submersible must be made of a specially formulated material, often a titanium alloy, and even then, the margins of safety are thin. A single flaw in the hull, a single miscalculation in the engineering, and the submersible would be crushed in an instant with no possibility of rescue. For these reasons, scientists have developed an alternative approach. They have built autonomous landers. These are simple steel frames, often no more than a few meters across, weighted with iron ballast. They are loaded with bait traps, cameras, and environmental sensors. Then they are dropped overboard from a ship. They sink under gravity, falling through the water column at a controlled rate until they reach the seafloor. Once there, they sit on the bottom, collecting samples and footage for 12 to 24 hours. The bait attracts scavengers, and the cameras record their behavior. The traps capture specimens for later study. The sensors measure temperature, salinity, oxygen, and other environmental variables. When the timer expires, the lander releases its ballast weights and the entire apparatus floats back up to the surface, where it is recovered by the ship. The elegance of this design is almost poetic. There are no thrusters, no engines, no complex propulsion systems. The lander uses gravity to descend and buoyancy to ascend. It is a passive instrument, a silent observer that sits on the seafloor and waits. And because it is unmanned, it does not require the expensive life support systems, the safety equipment, or the crew that a submersible demands. A lander can be built for a fraction of the cost of a crewed submersible, and it can be deployed repeatedly, year after year, from a wide range of ships. This has democratized Hadal research. It has allowed scientists from institutions that could never afford a submersible to study the deepest parts of the ocean. And it has produced a wealth of data that was simply unavailable before. One of the most important projects using landers is the Hades program, the Hadal Ecosystem Studies, led by Jeff Drazen and Patricia Fryer at the University of Hawaii. The project was funded by the United States National Science Foundation beginning in 2012, and it has been at the forefront of Hadal research ever since. Hades has deployed landers in the Mariana Trench, the Kermadec Trench, and other Hadal environments, and it has transformed our understanding of these ecosystems. The landers have revealed that the Hadal Zone is not a barren, lifeless desert. It is teeming with life, from tiny microbes to large fish and crustaceans. The species that live there are often unique to the trenches, having evolved in isolation over millions of years. And the food webs that sustain them are complex and dynamic, driven by the slow rain of organic matter from the surface and by the occasional catastrophic input of large food falls, such as whale carcasses. What the landers have revealed is nothing short of revolutionary. We used to think of the deep ocean as a place of slow, steady, unchanging conditions. We thought of it as a refuge from the variability of the surface. But the landers have shown that the Hadal Zone is a dynamic environment, subject to currents, storms, and even earthquakes. The trenches are often located at subduction zones, where tectonic plates collide and one plate is forced beneath another. These are regions of intense geological activity. The seafloor is constantly shifting, and the sediment is periodically disturbed by turbidity currents and landslides. The organisms that live there must be adapted not only to extreme pressure, but also to a highly variable and sometimes violent environment. Among the most remarkable discoveries of the lander program is the Hadal snailfish. The species Pseudoleparus swerii was formally described by Thomas Linley and his colleagues in 2017, based on specimens collected from the Mariana Trench at depths of 6,198 to 8,076 meters. The name honors Herbert Swire, a navigating sub-lieutenant on the HMS Challenger expedition, which conducted the first comprehensive survey of the deep sea in the 1870s. The snailfish is a small, pale, translucent creature. It has no scales. Its body is gelatinous and soft, almost like jelly. It has an elongated tail and a large head with small eyes. It is not a fearsome predator. It is a gentle scavenger, feeding on tiny crustaceans and other small organisms that live in the sediment. The gelatinous body of the snailfish is itself an adaptation. At the pressures of the Hadal Zone, any rigid structure would be compressed and crushed. A skeleton, a shell, a swim bladder, any of these would be a liability. The snailfish has done away with all of them. Its body is mostly water and jelly, and this allows it to equalize its internal pressure with the external pressure of the surrounding water. There is no pressure difference between the inside and the outside of the fish, so there is no force pushing in on it. The fish is, in effect, at equilibrium with its environment. This is the secret of its survival. It does not resist the pressure. It surrenders to it. It becomes one with the water around it. The snailfish is related to other common ocean fish. It belongs to the family Liparidae, which includes more than 400 species found in oceans around the world. Many of these species live in shallow water, in the cold seas of the Arctic and Antarctic. They are all characterized by their soft, gelatinous bodies and their lack of scales. The Hadal snailfish is simply the deepest living member of this family, the one that has pushed the boundaries of the fish body plan to its absolute limit, and it is not alone. Other species of snailfish have been found in other trenches, and they all share the same basic adaptations. The snailfish tend to cluster at certain depths. They are not distributed evenly throughout the Hadal zone. They are most abundant at depths of around 7,000 to 8,000 meters, and they become less common as the depth increases. This clustering is not random. It reflects the distribution of their food. The snailfish feed on small crustaceans, particularly amphipods, which are also more abundant at these depths. The snailfish have evolved to exploit this food source, and they are most successful where it is most plentiful. And then there is a depth beyond which the snailfish do not go. Below about 8,400 meters, the snailfish disappear. This is not a matter of food availability. The amphipods are still there. The temperature is still stable. The pressure is still just a physical force. But the snailfish are absent. This has led to a scientific debate about why this depth represents a biological ceiling for fish. One hypothesis is that the TMAO concentration required to counteract the pressure at these depths would be so high that it would exceed the limits of what the fish can tolerate. The TMAO itself would become toxic. Another hypothesis is that the proteins of the snailfish have reached the limit of their evolutionary flexibility. They cannot evolve to be more flexible, more resistant to pressure, without losing their ability to function at all. The line between life and death in this case is a fine one, and it is drawn at a depth of about 8,400 meters. The reason for this ceiling is still debated, and it is one of the open questions in Hadal biology. But the absence of snailfish below this depth does not mean that the deepest parts of the trench are lifeless. The dominant organisms in the deepest waters are not fish at all. They are amphipods. These are small, shrimp-like crustaceans that are found throughout the ocean, from the intertidal zone to the Hadal depths. In the Hadal zone, they are the dominant scavengers, the vultures of the deep. They are the ones that swarm to anything that falls from above, whether it is a fish carcass, a whale fall, or any other source of organic matter. The most famous of these Hadal amphipods is Hirandelia gigas, a species that dominates the Mariana Trench. It is a small creature, only a few centimeters long, but it is present in astonishing numbers. When the landers descend with their bait, they are often overwhelmed by swarms of Hirandelia gigas within hours. The amphipods detect the bait from a distance, using their highly sensitive chemical detection systems. They can smell the compounds released by decaying flesh at concentrations that are almost unimaginably low. They are drawn to the bait from all directions, and they arrive in a frenzy of feeding that can strip a carcass to the bone in a matter of hours. There is also a giant amphipod, Alicella gigantea, which can reach lengths of 34 centimeters. This is a truly enormous crustacean, as long as a human forearm, and it is a formidable scavenger. It was first discovered in the 19th century, but it was not until the era of lander exploration that we understood its true abundance and distribution. Alicella gigantea is found in the deepest parts of the ocean, and it is a reminder that the Hadal zone is not a place of tiny, fragile creatures. It is a place where life can reach remarkable sizes, even under the most extreme conditions. The amphipods are the foundation of the Hadal food web. They are the primary consumers of the organic matter that falls from above. They are also the prey of the snailfish and other predators. The relationship between the amphipods and the snailfish is one of the most important dynamic in the Hadal ecosystem. The amphipods are the engine that drives the system, converting the detritus of the surface into the biomass that sustains the entire community. The chemical detection systems of the amphipods are extraordinary. They are among the most sensitive in the animal kingdom. They can detect the odor of a food source at distances of hundreds of meters, perhaps even kilometers. This is essential in an environment where food falls are rare and unpredictable. A whale carcass that sinks to the bottom of the Mariana Trench is a once-in-a-lifetime event for the organisms that live there. It is a bonanza, a feast that must be exploited quickly before it is consumed by others. The amphipods have evolved to detect these events with remarkable precision, and they can mobilize in enormous numbers within a short time. This is why the landers are so effective at capturing them. The bait is a signal, and the amphipods respond to it with a speed and efficiency that is almost frightening. But the contamination of the Hadal zone by human activity is a growing concern, and it is a story that is still unfolding. The plastic debris that we see floating on the surface of the ocean, the chemical pollutants that we release into the air and water, the radioactive isotopes that we have generated through nuclear testing, all of these are finding their way into the deepest parts of the ocean. The Hadal Zone is not isolated from the rest of the planet. It is connected to the surface by the constant rain of particles, by the currents that circulate through the deep, and by the food falls that bring organic matter from the sunlit waters above. The pollutants that we release at the surface are being transported to the trenches, and they are accumulating in the tissues of the organisms that live there. The amphipods, the snailfish, and the microbes of the Hadal Zone are all being exposed to these human-made substances, and the consequences are not yet fully understood. When we measure the concentration of TMAO in a deep-sea snailfish, we are also measuring the presence of other chemicals that we have put there, and that is a troubling thought. The deepest parts of the ocean are not pristine. They are not a refuge from the Anthropocene. They are a sink, a place where the detritus of our civilization accumulates, and the organisms that live there are bearing the burden of our actions. We are only beginning to understand the full extent of this contamination, and the implications for the future of the Hadal Zone are uncertain. The data that we have collected so far, from the landers and the submersibles, suggest that the problem is more severe than we expected. Microplastics have been found in the guts of Hadal amphipods, and persistent organic pollutants have been detected in the tissues of deep-sea fish. The contaminants are not evenly distributed. They are concentrated in certain regions, particularly in the trenches that are closest to industrialized coastlines. The Mariana Trench, for all its remoteness, is not immune, and the organisms that live there are being exposed to a cocktail of chemicals that did not exist when they first evolved. The long-term effects of this exposure are unknown, but the early signs are not encouraging. We are watching, in real time, as the last pristine environment on Earth becomes contaminated, and we are struggling to understand what this means for the creatures that call it home. The Hadal Zone is not infinite. It is not a bottomless pit. It is a finite ecosystem with limited resources and limited capacity to absorb the insults that we are inflicting upon it. And as we continue to study it, as we continue to send our landers and our submersibles into its depths, we are confronted with a paradox. We are learning more about the Hadal Zone than ever before, but we are also learning that it is changing, and that the changes are driven by forces that are far beyond the control of the organisms that live there. The snailfish that swims at 8,000 meters, the amphipod that scours the seafloor for food, the microbe that thrives in the sediment, they are all connected to us, and we to them. The contamination of the Hadal Zone is not a remote problem. It is a problem that we are all part of, and it is a problem that will not be solved by simply ignoring it. The deep ocean is not a separate world. It is a part of our world, and the decisions that we make at the surface will shape the fate of the creatures that live in the deepest parts of it. We have already begun to see the effects, and the question is not whether the Hadal Zone will be affected by our activities. It is whether it will survive them. The answer to that question is still unknown, and it depends on choices that we have not yet made. But as we continue to explore the Hadal Zone, as we continue to send our instruments into its depths, as we continue to measure the TMAO in the snailfish and the plastics in the amphipods, we are writing a story that we cannot yet read. The fate of the Hadal Zone is intertwined with our own, and the contamination that we are now detecting is a signal, a warning that we are not separate from the deep ocean, that we are part of it, and that the consequences of our actions are being felt in the most remote places on Earth. The data are accumulating, the measurements are being made, and the landers are still descending, and the story is still being written, and we are only beginning to understand what it means. Consider for a moment what it means to measure a chemical that was banned before you were born, in a place that no human had ever seen until a few years ago. Alan Jameson, a marine biologist at Newcastle University, had spent his career studying the Hadal Zone, the deepest region of the ocean, and he had begun to notice something that troubled him deeply. When his team published their findings in the journal Nature, Ecology, and Evolution in 2017, the scientific community was forced to confront an uncomfortable truth. The amphipods they had collected from both the Mariana Trench and the Kermadec Trench, creatures living more than 10,000 meters below the surface, were carrying a chemical burden that should not have been possible. The compounds in question were polychlorinated biphenyls, known as PCBs, and polybrominated diphenyl ethers, known as PBDEs. These are synthetic chemicals that were manufactured in vast quantities throughout the 20th century. PCBs were used in electrical equipment, in hydraulic fluids, in heat transfer systems, and in 1,000 other industrial applications. They were prized for their stability, for their resistance to heat and pressure, for their remarkable ability to survive almost any environmental condition. That stability, which made them so useful in industry, is precisely what makes them so dangerous in nature. When PCBs were finally banned in the 1970s and 1980s in most countries, after decades of evidence linking them to cancer and neurological damage, the chemicals did not simply disappear. They remained in the environment, cycling through the air, the water, the soil, and the living tissue of every organism they encountered. What Jameson and his colleagues found was staggering. The concentration of PCBs in the amphipods of the Mariana Trench was 50 times higher than the concentration found in crabs from polluted rivers in China, rivers that flow through some of the most industrialized landscapes on Earth. For context, crabs from those rivers had already been identified as among the most contaminated crustaceans ever measured. To find levels 50 times higher in animals living 11 kilometers below the surface, in a place that most people still imagine as pristine and untouched, was a revelation that reshaped our understanding of how pollution travels through the global environment. The pathway by which these chemicals reach the Hadal zone is a story of slow, patient descent. It begins at the surface, where PCB molecules released decades ago into the atmosphere eventually washed into the ocean with rainfall. Once in the marine environment, these hydrophobic compounds, chemicals that repel water, immediately sought out surfaces to cling to. They adsorbed onto marine particles, onto the tiny fragments of organic detritus that drift through the surface waters, onto the fecal pellets of zooplankton, onto the bodies of dead phytoplankton, sinking slowly toward the abyss. Each particle carried its tiny burden of contamination, and each particle began its long journey downward. The sinking process is not rapid. A particle leaving the surface can take weeks, months, or even years to reach the Hadal depths. Along the way, bacteria in the water column consume the organic matter, breaking it down and releasing much of its energy, but the PCBs remain untouched. They are persistent, which is their defining characteristic. They do not degrade. They do not dissolve. They simply travel, clinging to whatever surface they have found, descending meter by meter through the darkness until they reach the bottom. And there, in the Hadal zone, they accumulate. The Hadal zone acts as a trap. The trenches of the deep ocean are not merely deep furrows in the seafloor. They are geological structures that actively funnel organic material downward. The steep walls of the trenches collect sediment and detritus from the surrounding abyssal plain, channeling it into the narrow trench floor. Anything that sinks through the water column above a trench is likely to end up in the trench itself. This is why the amphipods of the Hadal zone, creatures that feed on whatever organic matter reaches them, are so heavily contaminated. They are at the bottom of a funnel that has been collecting the chemical legacy of the 20th century for decades. The irony is almost unbearable. The most remote places on Earth, the deepest points of the ocean, the locations that we have only recently begun to explore, now carry the unmistakable chemical signature of industrial civilization. The PCBs that were manufactured in factories in the United States, in Europe, in Japan, that were released into the atmosphere and the oceans decades ago, have found their way to the bottom of the Mariana Trench. They have been consumed by amphipods that have never seen sunlight, that have never encountered a human being, that have evolved in complete darkness and crushing pressure for millions of years. And yet they carry within their fatty tissue the legacy of our factories, our power plants, our discarded electrical equipment, our careless disposal of the things we no longer wanted. The PBDEs found in the same amphipods tell a similar story. These flame retardants were added to furniture, to electronics, to textiles, to countless consumer products throughout the late 20th century. When those products were disposed of, when they were burned in landfills or simply discarded, the PBDEs entered the environment. They followed the same path as the PCBs, adsorbing onto particles, descending through the water column, and accumulating in the fatty tissues of deep-sea organisms. The fact that both classes of chemicals were found together in the Hadal amphipods is not a coincidence. It is a signature of the industrial era, written into the bodies of creatures that have never seen the world that produced these chemicals. The implications of this discovery extend far beyond the amphipods themselves. When these contaminated creatures are consumed by larger predators, the chemicals move up the food chain. Each successive level concentrates the toxins further, a process known as biomagnification. The Hadal zone is not a dead end for these chemicals. It is a reservoir, a storage facility, a place where they accumulate and persist, waiting to be released into the broader ecosystem. The fish that migrate from the Hadal zone to shallower waters, the scavengers that feed on the carcasses of Hadal organisms, all of them become vectors for this contamination. Now consider the second major contamination story that emerged from the Hadal zone. On the 1st of May 2019, Victor Vescovo, a retired United States Navy officer and private explorer, descended to the bottom of the Challenger Deep in the Mariana Trench. His submersible, the DSV limiting factor, reached a depth of 10,928 meters, a record that would stand as the deepest manned dive in history. And there, on the seafloor, in the darkness of the deepest place on Earth, he found a plastic bag. He also found sweet wrappers. These items, discarded perhaps decades ago, perhaps only years ago, had made the same journey as the PCBs. They had sunk through the water column, had been carried by currents, and had come to rest at the bottom of the world's deepest ocean. The discovery of plastic at Challenger Deep was not entirely unexpected. A 2020 study, conducted by researchers who analyzed samples from 6 different trenches, found microplastics in the digestive systems of Hadal amphipods from every single location. The microplastics were tiny fragments, most smaller than a millimeter, but they were present in every animal examined. The mechanism by which plastic reaches the Hadal floor is similar to the mechanism that carries PCBs. Plastic debris at the surface, whether from discarded bags, from fishing gear, from the millions of tons of waste that enter the ocean every year, gradually breaks down into smaller and smaller pieces. Sunlight and wave action fragment the larger pieces, and these fragments, along with the fibers shed from synthetic clothing and the pellets used in industrial manufacturing, become part of the marine snow that falls through the water column. Some plastic sinks because it is denser than seawater. Some plastic sinks because it becomes covered with marine organisms, with algae and bacteria that add weight to its surface. Some plastic is consumed by marine animals, which then excrete it in their fecal pellets, and those pellets sink. Whatever the mechanism, the plastic reaches the bottom, and there it accumulates. The Hadal zone, which we have already seen acts as a trap for organic material and contaminants, is equally effective at trapping plastic. What happens to the amphipods that consume this plastic? The studies have shown that the plastic fragments are present in their digestive systems and that they are not expelled. The plastic remains in the gut, taking up space that could be used for actual food, potentially damaging the digestive tract, and possibly releasing chemical additives into the animal's tissues. The plastic itself is a vehicle for contamination. The additives used in plastic manufacturing, such as bisphenols and phthalates, are known endocrine disruptors, and they can leach from the plastic into the animal's body. The hydrophobic contaminants that we discussed earlier, the PCBs and PBDEs, also adsorb onto the surface of plastic fragments, meaning that each piece of microplastic is a tiny delivery system for a cocktail of toxic chemicals. The Hadal amphipods, these small crustaceans that look like pale, translucent shrimp, are living at the intersection of 2 of the most significant environmental challenges of our time. They are consuming plastic that has descended from the surface, and they are accumulating the chemical contaminants that have made the same journey. They are the canaries in the coal mine of the deep sea, and their bodies are telling us a story that we are only beginning to understand. But how do these creatures survive at all? The Hadal zone, which we think of as the most food-limited environment on Earth, is home to a surprising abundance of life. The question of how anything can survive at 11,000 meters, where there is no sunlight, no photosynthesis, and where the food supply is a slow trickle of organic matter from the surface, has puzzled scientists for decades. The answer lies in a phenomenon that is both simple and profound: marine snow. Marine snow is the continuous, gentle fall of organic material from the surface waters of the ocean. It consists of dead phytoplankton, of fecal pellets from zooplankton, of the mucus and detritus that are produced by the countless organisms that live in the sunlit upper layers of the sea. This material falls slowly, drifting downward at rates that vary from a few meters per day to a few hundred meters per day, depending on the size and density of the particles. A particle that leaves the surface near the equator, where the water is warm and the biological productivity is high, may take weeks or months to reach the Hadal depths. Along the way, it is consumed by bacteria, by zooplankton, by the countless organisms that live in the water column. Each level of consumption removes some of the organic matter, and by the time the particle reaches the Hadal zone, most of its caloric value has already been extracted. The Hadal zone is therefore a very poor environment. The food that arrives is scarce, and it is mostly in the form of partially decomposed organic matter, the scraps of the scraps of the scraps. Yet the trenches are not barren. They are home to a remarkable diversity of life, from amphipods to holothurians, from polycate worms to isopods, from fish to crustaceans. How do they survive? The answer lies in the geological structure of the trenches themselves. Alan Jameson and his colleagues described this in a concept they called the geological amplifier. The trenches are not simply holes in the seafloor. They are elongated depressions formed by the subduction of one tectonic plate beneath another. They are typically steep-sided, with walls that rise at angles of up to 30 degrees or more. These walls act as funnels, collecting organic material that falls from the water column above the surrounding abyssal plain. Any organic matter that sinks within the catchment area of the trench is channeled down the sloping walls, accumulating on the trench floor. The result is that the Hadal zone, despite being the most food-limited environment imaginable, is paradoxically richer in organic material than the surrounding abyssal plains. This is the geological amplifier in action. The shape of the trench concentrates the meager food supply, amplifying whatever organic material arrives from the surface. The effect is not enormous, but it is significant. Studies have shown that the sediment at the bottom of trenches contains higher concentrations of organic carbon than the sediment of the adjacent abyssal plains. The amphipods, the worms, the crustaceans that live in the trenches are able to survive because their environment provides just enough food, and because they have evolved to be extraordinarily efficient at extracting energy from the limited resources available. But there are also pulses of food that arrive at the Hadal zone in much larger quantities. Consider the whale fall. When a large cetacean, a whale, dies and sinks to the bottom of the ocean, its carcass represents an enormous pulse of organic material. A blue whale can weigh over 100 tons, and its body contains a vast amount of energy-rich tissue. When such a carcass reaches the Hadal depths, it becomes a feast for the scavengers of the deep. The amphipods, the isopods, the hagfish, and the other scavengers that live in the trench will consume the carcass over a period of months or years, feeding on the soft tissue and the bones. The whale fall is a rare event, but when it happens, it provides a concentrated source of food that can sustain a large population of Hadal organisms for a long time. Similarly, there are woodfalls. When a large tree falls into the ocean, or when a log from a river reaches the sea, it can become waterlogged and sink. If it sinks in the vicinity of a trench, it will be carried down the slope and deposited on the trench floor. Wood is not a particularly nutritious food source, but it harbors a community of organisms, including bacteria and fungi that break down the cellulose, and these organisms are consumed by the larger scavengers of the Hadal zone. The woodfall provides a habitat as well as a food source, and it can support a diverse community of life for many years. The combination of marine snow, whale falls, and woodfalls provides the Hadal zone with a food supply that is sparse but sufficient. The organisms that live there have evolved to take advantage of every available resource. They have slow metabolisms, allowing them to survive on very little food. They have large mouths and expandable stomachs, allowing them to consume large quantities of food when it becomes available. They are adapted to a life of feast and famine, and they are remarkably successful at it. The Hadal food web is a delicate balance of predators and prey, of scavengers and decomposers, of organisms that are adapted to the most extreme conditions on Earth. The amphipods are the most abundant animals in the Hadal zone, and they occupy a central position in the food web. They feed on the organic detritus that reaches the trench floor, and they are in turn consumed by the larger predators, such as the Hadal snailfish, which have been observed at depths of over 8,000 meters. The snailfish, in turn, are preyed upon by the larger fish and crustaceans that venture into the Hadal zone. It is a complex web of interactions, and it is a web that we are only beginning to understand. The study of the Hadal food web is one of the many scientific goals of the expeditions that have been conducted in recent years, and among the most significant of these expeditions was the 5 Deeps expedition, led by Victor Vescovo. Vescovo, a retired United States Navy officer and a private explorer, had a vision that was both ambitious and audacious. He wanted to dive to the deepest point in each of the 5 oceans, and he wanted to do it in a specially designed submersible that could withstand the crushing pressures of the Hadal Zone. The submersible, the DSV limiting factor, was built by Triton Submarines, a company based in Florida. Its construction cost approximately $48 million, a figure that reflects the extraordinary engineering challenges involved. The pressure hull, made of titanium, was designed to withstand pressures of over 11,000 meters. The thick acrylic viewport, the glass that would allow Vescovo and his pilots to see the Hadal Zone, was the result of years of research and development. The submersible was designed to be resettable, meaning that it could be used multiple times, and it was equipped with a range of scientific instruments, including cameras, lights, and a robotic arm for collecting samples. The 5 Deeps expedition began in December 2018, with a dive to the bottom of the Puerto Rico Trench in the Atlantic Ocean. The Puerto Rico Trench is the deepest point in the Atlantic, reaching a depth of 8,376 meters. Vescovo and his team descended to the bottom, where they observed the seafloor and collected samples. The trench, which lies just north of the Caribbean, is a particularly interesting location because it is influenced by the outflow of the Amazon River, which carries vast amounts of sediment and nutrients from South America. The sediment in the trench is thick, and the biological community is relatively rich, reflecting the input of organic material from the river. In February 2019, the expedition moved to the South Sandwich Trench, the deepest point in the Southern Ocean, at a depth of 7,434 meters. This trench is located in the remote South Atlantic, near the islands of South Georgia and the South Sandwich Islands, and it is notable for its extreme isolation. The dive was conducted in the middle of the southern summer, but the conditions were still challenging, with strong winds and rough seas. The bottom of the South Sandwich Trench was found to be home to a variety of organisms, including amphipods and holothurians adapted to the cold, dark conditions. In April 2019, the expedition dived to the Java Trench, the deepest point in the Indian Ocean, at a depth of 7,192 meters. The Java Trench, also known as the Sunda Trench, lies south of Indonesia, and it is an area of intense seismic activity. The dive was conducted in challenging conditions, and the team encountered some difficulties with the submersible, but they were ultimately successful. The bottom of the Java Trench was found to be covered with a thick layer of sediment, and the biological community was relatively sparse, reflecting the limited input of organic material. The most significant dive of the expedition came in May 2019, when Vescovo descended to the bottom of the Challenger Deep in the Mariana Trench, reaching a depth of 10,928 meters. This was the deepest point on Earth, and the dive set a new record for the deepest manned descent. Vescovo spent 4 hours on the bottom, exploring the seafloor and collecting samples. He observed the plastic bag and the sweet wrappers that we mentioned earlier, and he collected sediment samples that would later be analyzed for their chemical and biological content. The dive was a success, and it provided the scientific community with a wealth of data about the deepest environment on Earth. In August 2019, the expedition completed its final dive, descending to the bottom of the Malloy Deep in the Arctic Ocean, at a depth of 5,150 meters. The Malloy Deep is the deepest point in the Arctic, and it is located in the Fram Strait between Greenland and the Svalbard archipelago. The dive was conducted in an area covered by ice, and the team had to use a specially designed icebreaker to keep the lead open. The bottom of the Malloy Deep was found to be relatively barren, with a thin layer of sediment and few organisms, reflecting the low productivity of the Arctic waters. The 5 Deeps expedition was a remarkable achievement. It was the first time that all 5 of the deepest points in the world's oceans had been visited by a single expedition, and it generated a vast amount of scientific data. The expedition also demonstrated the value of private exploration, showing that a well-funded individual with the right resources and the right team could accomplish what had previously been the domain of national space agencies and government-funded research programs. The scientific value of having a private expedition with the resources to actually attempt all 5 dives was immense, and it opened up new possibilities for future exploration. The samples collected during the 5 Deeps expedition, and during the many other expeditions that have visited the Hadal Zone in recent years, are being analyzed in laboratories around the world. The sediment samples, the water samples, the biological samples, all of them are yielding new insights into the mysteries of the deep. And among the most powerful of the new techniques being used to study these samples is environmental DNA, or eDNA. Environmental DNA is a remarkable technology. It is based on the simple observation that all organisms shed DNA into their environment. As fish swim, they release cells into the water. As amphipods feed, they leave traces of their genetic material. As bacteria multiply, they release fragments of their DNA into the surrounding water. These DNA fragments are everywhere, and they can be collected and analyzed. Water samples from the Hadal zone, when filtered and processed, reveal the genetic signatures of all the organisms that have recently been present in that water. The technique has been used to study the Hadal zone in a way that was previously impossible. In the Mariana Trench, eDNA surveys have revealed the presence of species that were never physically captured. The water samples contain genetic traces of fish, of crustaceans, of worms, of organisms that have never been seen by human eyes. The eDNA tells us that these species are present, that they are living in the Hadal zone, feeding and reproducing, even though we have never collected a single specimen. The gap between detecting that something is there and actually knowing what it does is one of the fundamental challenges of Hadal research. The eDNA tells us that a species is present, but it does not tell us what that species eats, how it reproduces, how it interacts with its environment, or how it is affected by the contaminants that we have found in the Hadal zone. To answer those questions, we need to physically capture the organisms, to observe them in their natural habitat, to study them in the laboratory, and that is far more difficult. The physical collection of Hadal organisms is an extraordinary challenge. It requires specialized equipment, such as baited traps that can be deployed from a surface ship and left on the seafloor for hours or days. The traps must be designed to withstand the immense pressure of the Hadal zone, and they must be constructed from materials that are not contaminated with the chemicals that we are trying to study. The organisms that are captured must be brought to the surface slowly to allow them to acclimate to the changing pressure, and they must be processed quickly to prevent their tissues from degrading. The eDNA surveys, the physical collections, the sediment samples, the water samples, all of these techniques are providing us with a picture of the Hadal zone that is more complete than ever before. But the picture is still incomplete. There are vast areas of the Hadal zone that have never been explored, deeps that have never been visited, organisms that have never been seen. The Hadal zone is the largest and least explored habitat on Earth, and we are only beginning to scratch the surface. The communication and engineering challenges of Hadal research are immense. The pressures at the bottom of the Mariana Trench are more than 1,000 times the pressure at the surface, and they pose a constant threat to any equipment that is deployed. The submersibles that dive to the Hadal zone must be built to withstand these pressures, and their pilots must be trained to operate in conditions that are unlike anything else on Earth. The communication systems that connect the submersible to the surface ship are limited, and the data that can be transmitted in real time is restricted. Most of the data collected during a Hadal dive is stored on the submersible itself and must be retrieved when the submersible returns to the surface. The engineering challenges are so significant that each dive is a major undertaking, and the planning that goes into a single dive can take months or years. The science that can be conducted during a single dive is limited, and the researchers must be extremely selective about what they choose to collect and study. The data that is collected, whether it is the sediment samples, the water samples, the biological specimens, or the video footage, is the result of a carefully planned sequence of operations, each of which must be executed with precision and precision alone. We are, in effect, blind when we work down there. The visible spectrum of light, which we rely on so instinctively, is absorbed by seawater within a few hundred meters. Beyond that, the world is a place of absolute darkness. And what of radio waves? They are the invisible threads of our modern world, but they are useless in the ocean. They are conductive, and seawater, being a conductive medium, acts as a shield against them. The signals we use to communicate with our satellites and our phones are simply swallowed by the immense body of the water. This leaves us with one tool, one single vector for information, and that is sound. Sound travels through water in a way that is almost alien to our terrestrial experience. It is not a waveform that we hear in the air. It is a pressure pulse that can travel for thousands of kilometers. It is the fundamental language of the deep. In the Hadal zone, therefore, we must rely on acoustic communication. It is the only way to speak to the machines we send down. The technology is a sophisticated form of telephony, but it is a telephony that is profoundly slow. An acoustic modem, which is attached to our submersible and our surface ship, converts a digital signal into a series of sound pulses. These pulses are sent upward through the water column, traveling at roughly 1,500 meters per second. That is the speed of sound in seawater, a speed that is dependent on temperature, pressure, and salinity, but which remains remarkably constant. To understand the challenge this presents, we must consider the sheer scale of the depths. If we are operating at the bottom of the Mariana Trench at 11,000 meters, the signal must travel 11,000 meters up to reach us. That is a journey of approximately 7 seconds. But we are not just sending a signal, we are waiting for a reply. We send a command, we wait for the vehicle to respond, and we wait for that response to return. This is a round trip. That means for every single command we send to a vehicle at maximum depth, we wait for a minimum of 14 seconds and often 15 seconds before we receive any confirmation. It is not a conversation. It is a slow exchange of letters, each one taking a quarter of a minute to arrive. This delay is not just an inconvenience. It is a fundamental constraint that dictates how we operate. We cannot pilot a deep-sea vehicle in real time. We cannot see a cliff wall ahead and react to it. By the time we see the cliff wall, we are already at it. We cannot steer a robotic arm with a joystick, because the transmission delay would make every movement a guess. The vehicle is not a remote-controlled toy, it is an autonomous agent. Its computers are programmed to follow a predetermined flight plan, to hold a constant altitude above the seabed, and to react to the immediate environment. The human pilot on the surface is not a pilot in the traditional sense. They are a supervisor, a guardian who watches the telemetry and the slowly transmitted data, and who can only intervene in the most general of ways, issuing a command to stop or to ascend or to change a heading, and then wait for an eternity of 14 seconds to see if it was obeyed. This is where the acoustic positioning system becomes so vital. We need to know where our vehicle is, and we need to know it without the benefit of a visual sightline. The system used is often called ultra-short baseline, or USBL. This involves a transceiver mounted on the bottom of the surface ship. This transceiver sends a sound pulse, which is like a ping, down to the vehicle. The vehicle, in turn, has a transponder that waits for this ping and immediately replies with its own ping. The ship's transceiver receives this reply. The time taken for the round trip gives us the distance. But the USBL system is cleverer than that. By measuring the phase difference of the sound wave as it arrives at the multiple small elements of the transceiver array, it can calculate the precise angle of the incoming signal. Multiply the distance by the angle, and we have a position. We can know, to within a few meters, where our vehicle is in the vast, featureless expanse of the deep. It is a system of triangulation built entirely on sound. But the bandwidth is our most precious commodity. The acoustic signals we send can carry only a tiny amount of digital information. At Hadal distances, that rate is a few kilobits per second. To put that in perspective, a modern video call uses millions of kilobits per second. Our acoustic link is 1,000 times slower than the slowest dial-up internet connection from the 1990s. It is enough to transmit a status report, a depth and heading reading, the temperature of the water, the battery voltage. It is enough to transmit a single, low-resolution still image, which might take a minute to stitch together. It is not enough to transmit video. The video that the submersible captures, the most important scientific data we will gather, is recorded on internal hard drives. It is stored in the cold and the dark, on a machine that knows not to fail. It is only when the vehicle returns to the surface, after its long journey of ascent, that we can retrieve this footage. We work in the blind, guided by a whisper of sound, and we must wait for the return to see the truth. This dance of sound and silence is not just a technical detail. It is the very fabric of how we explore. It shapes our psychology, the patience required of every scientist on board. It is this patience that was so tested in the aftermath of one of the most powerful geological events ever recorded. On the 11th of March 2011, a massive earthquake, a magnitude 9.0, struck off the coast of Tohoku, Japan. The epicenter was in the Japan Trench, a deep and ominous scar in the ocean floor that runs offshore of the island of Honshu. This trench is not as deep as the Mariana, but it reaches depths of over 9,000 meters. It is a subduction zone, where the Pacific Plate is diving beneath the North American Plate. The earthquake, one of the largest ever recorded by our instruments, shifted the seafloor by tens of meters. It also triggered a devastating tsunami, But it also did something else. It triggered a series of massive submarine landslides along the steep walls of the trench. For years, the scientific community of Japan, through the Japan Agency for Marine Earth Science and Technology, or JAMSTEC, has been a pioneer in hadal research. They have been exploring the depths since the 1980s, with their own fleet of submersibles and remotely operated vehicles. They were, therefore, uniquely positioned to study the aftermath of this disaster. They deployed their ROV, Kaiko, a vehicle that had already made history by being the first to reach the bottom of the Mariana Trench, and later their crewed submersible, the Shinkai 6500, which can carry 3 people to a depth of 6,500 meters. These were not simple missions. They were intricate, slow operations, using the acoustic communication we have described. They had to send sound pulses down into the trench, listen for the replies, and slowly guide their vehicles to the seabed. What they found was a world that had been torn apart. The sediment on the trench floor was a chaotic mess, a slurry of mud and rock that had been churned up by the landslides. The seafloor topography, which had been mapped before, had been completely altered. Canyons were filled, ridges were flattened, and in some places the floor had been lifted up. But the most profound discoveries were about the life there. The communities of organisms that had lived on the trench floor, the scavenging amphipods and the deposit-feeding worms, had been buried. The earthquake had reset the clock. The trench was a place of mass extinction, a localized catastrophe. But the JAMSTEC scientists did not just observe the destruction. They watched the recovery. They returned to the same sites year after year in the months and years that followed. The vast majority of the seafloor in the Hadal Zone is a food-poor desert. The animals that live there are adapted to a life of scarcity, waiting for the occasional unpredictable fall of food from the surface. The earthquake, however, had mixed the steep slopes of the trench, and this mixing had brought a sudden influx of organic matter. The sediment that had been buried was now exposed. The initial results were a barren wasteland. But then, the opportunists arrived. The amphipods, the hardy scavengers of the deep, were the first to return, their numbers exploding in response to the new food source. The recovery is a slow process, a story of life slowly re-establishing its foothold in a place of utter hardship. It is a story that we can only read through the lens of sound, a story that is still being written in the cold, dark sediments of the Japan Trench. Our story now moves to a different ocean, a different plate boundary. The Atlantic Ocean is home to its own Hadal Trench, the Puerto Rico Trench. It is the deepest point in the Atlantic, reaching a maximum depth of 8,376 meters at a place known as the Milwaukee Deep. This trench is a different kind of geological feature. It sits near the boundary between the Caribbean Plate and the North American Plate, and its formation is complex, a result of the North American Plate diving beneath the Caribbean Plate. It is a place of immense geological pressure, but it is also a place of profound biological difference. The Hadal fauna of the Atlantic is not the same as the Hadal fauna of the Pacific. This is due to a simple yet powerful fact of geography. The Atlantic and Pacific trenches are separated by vast continental landmasses and by the shallow waters of the mid-ocean ridges. The deep, cold, and isolated waters of the Hadal Zone are not a highway for dispersal. They are a series of isolated islands, separated by vast expanses of inhospitable water. The Pacific trenches have a long, complex history, and they are connected to each other by the deep currents of the Southern Ocean. The Atlantic, however, is more isolated. Its Hadal fauna has evolved independently for a long period of time. The species that live there are not the same as those in the Mariana. They are a different set of animals, a different set of evolutionary solutions to the same problem. The Puerto Rico Trench has been relatively understudied compared to the Pacific systems. This is a simple matter of access and focus. The Pacific has received the lion's share of our attention because it is home to the deepest points on Earth. The Atlantic, however, is a scientific frontier in its own right. The species that have been found there, the snailfish, the amphipods, and the unusual isopods, are just as fascinating as their Pacific cousins. They have adapted to the same extreme conditions, but their genetic lineage is their own. They represent a different branch of the tree of life, a branch that we have only just begun to explore. The Milwaukee Deep is a place of silence, but it is also a place of unique biodiversity, a biological treasure chest that is still waiting to be fully opened. To the south, in the Pacific Ocean, we find the Tonga Kermadec system. This is the second deepest trench system on Earth, a vast and complex feature that reaches a maximum depth of 10,182 meters in the Tonga Trench. It lies to the east of New Zealand and the island of Tonga in the South Pacific. It is a place of immense geological activity, a subduction zone where the Pacific Plate is diving beneath the Indo-Australian Plate. Because of its location, it is a crucible of life. The South Pacific trenches are not the same as the Mariana, despite their similar depths. They are subject to different currents, different food inputs, and a different geological history. The surface waters above them are more productive, meaning there is more organic matter falling to the deep, which in turn supports a richer and more numerous community of life. One of the most remarkable discoveries in this system came from a recent expedition, a result of a collaboration between researchers from the University of Western Australia and the Tokyo University of Marine Science and Technology. In 2023, they were exploring the Izu Ogasawara Trench, which is a nearby trench, not the Tonga itself. They were using a drop camera system, a lander that descends to the seabed and films the life that comes to investigate its bait. The video they captured, which was stored on the lander's internal hard drive and retrieved after its ascent, showed a group of snailfish. These are not the fish we see in our rivers. They are pale, translucent creatures, perfectly adapted to the crushing pressure. The researchers identified these fish as what they believe to be Pseudoliparus belyaevi, a species of snailfish. But the most important detail was the depth. The camera was on the seabed at 8,136 meters. This was the deepest fish ever observed on camera. It was a record, but more than that, it was a confirmation. It showed that these fish, these delicate-looking creatures, can thrive at depths that were once thought to be impossible for fish to survive. The Kermadec Trench, a part of this same system, has also been the subject of a famous study. It was here that a team of scientists, led by Alan Jameson, examined the contaminants in the amphipods that live in the deepest parts of the trench. These are the same scavenging crustaceans that we have spoken of, the ones that dominate the Hadal food chain. They are the cleanup crew of the deep, but they are also a warning. The study found that the amphipods in the Kermadec Trench contained high levels of persistent organic pollutants, harmful chemicals that are manufactured by humans and have been banned in many countries. These chemicals are not soluble in water, so they do not sink. Instead, they are carried by the surface currents, and they enter the food chain. They are consumed by plankton, which are consumed by small fish, which are consumed by larger fish. When these animals die, their bodies descend to the deep. The amphipods, scavenging on this falling detritus, are consuming a concentrated dose of human history. The Kermadec Trench, a place that is remote and pristine in our imagination, is a mirror of our own pollution. It is a powerful reminder that nowhere on Earth is truly isolated. This brings us to the fundamental question of cost. The Five Deeps expedition, which was a private, philanthropic effort to visit the deepest point in each of the 5 oceans, cost approximately $31 million. This is a sum that is available to a wealthy individual, but it is a sum that far exceeds the annual research budgets of most national oceanographic programs. The entire field of Hadal science is chronically underfunded relative to its scientific importance. We spend billions of dollars on space exploration, sending robotic probes to the far reaches of our solar system, but we have sent a fraction of that money to the bottom of our own oceans. The reasons are complex. Space is a frontier that captures our imagination, a symbol of human achievement. The deep ocean is dark, hidden, and difficult to sell to the public. It is out of sight and therefore out of mind. The results of Hadal science are slow and small in quantity. We do not discover a new planet every year. We discover a new species of amphipod, which is a far less exciting headline. The difficulty of making the case to funding agencies is immense. A research proposal for a Hadal expedition must be meticulously planned, but it is a gamble. The weather may be bad, the equipment may fail, and the results may be sparse. The science is hard, and the rewards are not guaranteed. This is why the field has been sustained by a few key institutions and a few passionate individuals. It is also why the advent of cheaper, more autonomous technology is so important. The lander systems, which are simple frames with cameras and bait, are relatively inexpensive. They can be deployed from a ship of opportunity without the need for a full-scale research vessel. They are democratizing access to the deep. They are allowing more scientists from more countries to study the Hadal zone. The autonomous vehicles, which are programmed to follow a survey path, are also becoming more affordable. They are not as flexible as a robot operated by a human, but they are more reliable. They can do the slow, patient work of mapping and observation. We are slowly, painstakingly, building a picture of the Hadal world. It is a world of crushing pressure, of absolute darkness, and of a silence that is only broken by the sound of our own ping. We are learning about its geology, its chemistry, and its biology. We are learning about the ways in which life can survive, and even thrive, in the most extreme conditions on our planet. We are learning about the deep, slow currents that connect all the trenches, and the way that our own actions, even at the surface, can reach down to the bottom. But for every question we answer, we uncover a dozen more. The sediment that we have not yet sampled, the species that we have not yet seen, the microbial communities that we have not yet imagined. We are like the first astronomers, looking up at a sky full of stars, but we are looking down at a world that is just as vast, just as complex, and just as full of mystery. And so, like the first astronomers, we have turned our gaze downward, sending our instruments into the deepest places on Earth, where the darkness has given up glimpses of the life that persists there. We have sent our instruments down into the deepest places on Earth, and we have seen glimpses of what lives there. But for every moment of observation, there are countless hours of darkness that remain unseen, and for every creature we have photographed, there are entire communities that we have never even known to look for. The Hadal Zone, the realm of the ocean that lies below 6,000 meters, is a frontier that we have only begun to explore, and the more we learn, the more we understand how little we truly know. The numbers that define this ignorance are striking, and they deserve our quiet attention. The total area of the Hadal seafloor, the land beneath the deepest trenches, covers approximately 2.3 million square kilometers. That is a vast expanse, roughly the size of the entire continent of Australia, and it represents between 1 and 2% of all the ocean floor on our planet. Yet of this enormous area, we have directly sampled less than 1%. We have lowered our corers and our grabs into the sediment of the trenches on perhaps a few hundred occasions across the entire history of oceanography, and each of those samples covers only a tiny patch of the bottom, a small circle of mud that we can hold in our hands. The rest of that 2.3 million square kilometers has never been touched by any instrument we have sent down. It has never been seen by a camera, and it has never been sampled by a corer. It lies in permanent darkness, and it waits. When we do manage to observe the Hadal floor, we are often looking at a surface that appears almost barren to the untrained eye. But we have learned that the surface is only the beginning. The sediment beneath our feet, or beneath the hull of our submersible, is alive in ways that we are only beginning to understand. The organisms that live within the top meter of Hadal sediment are known as the infaunal community, and they are invisible to every camera we have ever deployed. They cannot be seen from a submersible window, and they cannot be captured by a net. They live their entire lives buried within the mud, and the only way we can ever know them is by taking a core sample, a long cylinder of sediment that we pull up from the depths and slice into thin layers for examination in a laboratory on the surface. What we find in those cores is remarkable, and it changes the way we think about life itself. The infaunal community of the Hadal Zone is dominated by creatures that we rarely think about and almost never see. Nematodes, the roundworms that are found in nearly every environment on Earth, thrive in the sediment of the trenches in extraordinary numbers. They are joined by foraminifera, single-celled organisms that build intricate shells around themselves, and by vast populations of bacteria that live in the spaces between the grains of sediment. When we count these organisms, the numbers are almost impossible to comprehend. Some studies have suggested that the sediment of the Hadal Zone contains more bacteria per cubic centimeter than any other deep-sea environment on Earth. A single teaspoon of mud from the bottom of the Mariana Trench may hold more living cells than there are people on the surface of our planet. This density of life is not a random accident. The Hadal trenches act as a kind of trap for organic material, a final resting place for the remains of everything that once lived in the waters above. When organisms die in the sunlit surface waters, they sink slowly toward the bottom, and much of their organic matter is consumed by scavengers along the way. But the trenches are so deep that they capture a disproportionate share of what remains. The walls of the trenches funnel material downward, and the sediment that accumulates on the Hadal floor is rich in organic carbon, a feast for the bacteria that live within it. These bacteria are engaged in a process that is of profound importance to our entire planet, and we are only just beginning to appreciate it. The decomposition of organic matter in Hadal sediment is a critical step in the global carbon cycle. When bacteria break down the remains of dead organisms, they consume oxygen and release carbon dioxide. But in the deep sediment of the trenches, something different happens. The sediment is so thick and the oxygen is so scarce that the decomposition is incomplete. Much of the organic carbon that falls into the trenches is not fully broken down. Instead, it is buried, layer upon layer, and locked away from the atmosphere for millions of years. This is one of the ways that our planet regulates its climate, and the Hadal zone is one of the most important places on Earth for this process. The bacteria that live in the Hadal sediment are not just surviving in an extreme environment. They are helping to keep the carbon that would otherwise contribute to global warming safely buried beneath the seafloor. But the infaunal community is not the only remarkable life that exists in the Hadal zone. There are organisms that live on the surface of the sediment that are so strange that they challenge our very definition of what an animal is. The xenophyophories are among the most extraordinary creatures on Earth, and they are found in the deepest parts of the ocean. They are single-celled organisms, protists, and they belong to a group that is neither plant nor animal nor fungus. They are something else entirely, and they can grow to a size that we would never expect from a single cell. Some species of xenophyophories, such as Syringamina fragilissima, can reach 20 centimeters across, the size of a dinner plate. They are the largest single-celled organisms on our planet, and they live in the most extreme conditions we know. How does a single cell grow to such a size? The answer lies in the way xenophyophories feed. They are deposit feeders, and they live by engulfing sediment particles and extracting the organic material that clings to them. They are enormous bags of protoplasm, and they slowly move through the sediment or extend their branched bodies across the seafloor, absorbing the nutrients they need. Their bodies are filled with a network of tubes and chambers, and they build their structure from the particles of sediment they consume, creating a skeleton that is as much a part of the environment as it is a part of the organism. When we see a xenophyophore on the Hadal floor, we are seeing a creature that has been alive for perhaps decades, slowly growing and feeding in the darkness. The xenophyophores play a vital role in the Hadal ecosystem. They are not just passive consumers of organic matter. They create structure on the seafloor, and their presence alters the environment around them. Their bodies provide a surface for other organisms to attach to, and their feeding activity mixes the sediment, aerating it and changing its chemistry. They are, in a sense, the gardeners of the Hadal zone, and they are so abundant that in some areas they cover the seafloor in a kind of living carpet. We have no idea how many species of xenophyophores exist, because we have only scratched the surface of the Hadal zone. But we know that they are there, and we know that they are ancient, and we know that they are essential to the functioning of the deepest parts of our ocean. The question of how life can survive at the pressures found in the Hadal zone is one of the most fascinating in all of biology. The pressure at the bottom of the Mariana Trench is more than 8 tons per square inch, a force that would crush a human instantly. And yet, life thrives there. The dominant form of life in the Hadal zone is microbial, and these microbes are known as piezophiles, or pressure-loving organisms. They have evolved to live in conditions that are unimaginable to us, and they cannot survive at the surface. If we bring a piezophilic bacterium up to the surface, it will die, because the pressure that is its natural environment is suddenly removed. These organisms are not just tolerant of high pressure, they require it. Their cellular machinery is built to function at pressures that would destroy any other form of life. The study of piezophilic bacteria is a relatively young field, and much of what we know comes from the work of a small number of dedicated scientists. One of the pioneers in this field is Douglas Bartlett, a microbiologist at the Scripps Institution of Oceanography, who began isolating and culturing piezophilic bacteria in the 1990s. Bartlett and his colleagues have spent decades developing the specialized equipment needed to study these organisms, and they have identified a number of genera that are found in the Hadal zone. Shewanella, Photobacterium, and Colwellia are among the most common, and each of them has evolved unique adaptations to life under extreme pressure. Their cell membranes are composed of different fatty acids than those of surface-dwelling bacteria, and their proteins are folded in ways that allow them to function at pressures that would crush the proteins of other organisms. The study of these microbes is not just an academic exercise. It has profound implications for our understanding of the origins of life on Earth, and for the possibility of life elsewhere in the universe. When we think about the search for life beyond our planet, we often think of Mars or of the icy moons of Jupiter and Saturn, but the environments that we find in the Hadal zone of our own ocean may be the best analogs for the conditions that exist elsewhere. The moon Europa, for example, is covered in a thick layer of ice, and beneath that ice lies an ocean that is many times deeper than any ocean on Earth. The pressure at the bottom of that hidden ocean would be enormous, and yet we have no reason to believe that life could not exist there. The same is true of Enceladus, a moon of Saturn that vents plumes of water into space, and of the subsurface oceans of other moons in the outer solar system. When we study the piezophilic bacteria of the Hadal zone, we are not just studying the life of our own planet. We are studying the conditions that life might experience in the most extreme environments of the solar system, and we are learning that life is far more resilient than we ever imagined. The megafauna of the Hadal zone, the creatures that we can see with our cameras, are often less abundant than the microbial life, but they are no less fascinating. Among the most common large animals on the Hadal seafloor are the holothurians, the sea cucumbers. These soft-bodied animals are related to starfish and sea urchins, and they are found in enormous numbers on the sediment of the trenches. Species such as Elpidia and Peniagone are particularly abundant, and they can be seen crawling slowly across the bottom in great numbers, their bodies often covering the seafloor in a kind of moving carpet. The sea cucumbers are deposit feeders, and they pass sediment through their digestive systems, extracting the organic material that is mixed in with the mud. In doing so, they are effectively plowing the Hadal floor, turning over the sediment and changing its structure. This process, known as bioturbation, is of great importance to the Hadal ecosystem. When sea cucumbers feed, they mix the sediment, bringing deeper material to the surface and burying surface material below. This mixing affects the way organic carbon is buried in the sediment, and it influences the chemistry of the seafloor. The sea cucumbers are not just the most visible animals on the Hadal floor. They are also among the most important agents of change, and their feeding activity shapes the environment in which all other Hadal life exists. We have only begun to understand the role that these animals play, and we have yet to observe many of their behaviors. But we know that they are there, and we know that they are essential to the functioning of the deepest parts of the ocean. Beyond the sediment, beyond the reach of our cores, there is a world that we are only just beginning to explore. The deep biosphere is the realm of life that exists below the seafloor, in the rock and sediment that lies beneath the Hadal zone. We have drilled into the ocean floor in many places, and we have found microbial life in sediment that is tens of meters below the seafloor surface. In some places, scientists have found evidence of life in rock that is kilometers below the bottom of the ocean. These organisms are not living in the dark, cold world of the sediment surface. They are living in the pores and cracks of the rock itself, surviving on the chemical energy that is released by reactions between the rock and the water that flows through it. They are a kind of life that we never imagined, an ecosystem that exists in the deepest recesses of our planet, and they may be the most abundant form of life on Earth. The deep biosphere is a humbling discovery. It suggests that the total biomass of life on our planet may be far greater than we ever thought. Some scientists have estimated that the deep biosphere may contain as much carbon as all the life on the surface combined. The bacteria and archaea that live below the seafloor are not just a curiosity. They are a major component of the Earth's living systems, and they are involved in cycling elements on a global scale. We are only just beginning to understand the deep biosphere, and we have no idea how many species it contains. But we know that it is there, and we know that it is vast, and we know that it is ancient. The tools we use to study the Hadal zone are evolving rapidly, and the future of Hadal research is one of the most exciting frontiers in all of science. For decades, the only way to reach the deepest parts of the ocean was in a crude submersible, a vessel that is expensive to build and dangerous to operate. But that is changing. Autonomous underwater vehicles, or AUVs, are becoming increasingly capable, and they are beginning to make Hadal research more accessible. The Nereid Under Ice, a vehicle developed at the Woods Hole Oceanographic Institution, is one of the most advanced examples of this new technology. It is designed to explore under ice, and it has been used to study the deep ocean in areas that were previously inaccessible. The Monterey Bay Aquarium Research Institute, known as MBARI, has also made significant contributions to hadal mapping, using advanced sonar and imaging systems to create detailed maps of the seafloor in the deepest trenches. These new technologies are not just making hadal research easier, they are making it possible for research institutions that could never afford a crude submersible to study the deep ocean. By 2030, it is possible that routine access to hadal depths will be achievable for a much wider range of scientists, and the knowledge we gain will be immense. We will be able to map the hadal seafloor in detail, to sample the sediment in more places, and to observe the creatures that live in the deepest parts of the ocean in ways that we have never been able to before. The future of hadal research is bright, and every new tool we develop brings us closer to understanding the mysteries of the deepest places on Earth. And yet, even with all these advances, we must remain humble. The hadal zone is vast, and our ability to explore it is still limited. Every time we send a vehicle into the depths, we see things that we have never seen before. And every time we bring a sample back to the surface, we find organisms that we have never identified. The more we learn, the more we realize how much we do not know. We are like explorers standing on the shore of a vast ocean, looking out at the horizon and wondering what lies beyond. We have sent our instruments down, and we have waited for the return. And we have seen glimpses of a world that is stranger and more beautiful than we ever imagined. But the hadal zone is not a place that gives up its secrets easily. It is a place of darkness and pressure, a place where life exists at the very limits of what we thought was possible. And the deeper we look, the more we realize that the deepest places on Earth are not empty wastelands. They are teeming with life, and they are full of mysteries that we are only just beginning to unravel. To understand this hidden life, we must first look to the terrain that supports it. Below 6,000 meters, the ocean floor is not a flat, featureless plain, but a series of deep, narrow gashes in the Earth's crust, trenches that slice down to depths that would crush a submarine like paper. These are the hadal zones, named for Hades himself, the Greek god of the underworld, and for decades we assumed they were nearly lifeless, too extreme for anything but the most resilient microbes. But we were wrong. We are always wrong about the deep sea, and that is precisely why we keep going back. The Hades Project, the Hadal Ecosystem Studies Program, is a consortium of scientists from the University of Hawaii, Woods Hole Oceanographic Institution, and the University of Southern Denmark. Jeff Drazen, Timothy Shank, and Ronnie Gludd lead this effort, funded by the National Science Foundation, and they have spent years building a picture of life in the trenches that challenges everything we thought we knew. Their approach is methodical, almost obsessive. They use standardized landers, free-falling metal frames equipped with cameras, baited traps, and sediment corers that descend to the bottom and sit there for hours or days, collecting data and samples before returning to the surface on command. The same landers are deployed across multiple trenches, from the Mariana to the Kermadec to the Japan Trench, so that the results can be compared directly, trench to trench, without the confounding variables of different equipment or different techniques. And what they have found is startling. The amphipods, those small shrimp-like crustaceans that thrive in the deepest waters, are more similar between trenches that are thousands of kilometers apart than they are to the amphipods living on the adjacent abyssal plains, the vast, flat expanses of the deep sea floor that surround the trenches. In other words, the trenches form their own biological province. A creature living in the Mariana Trench has more in common with a creature living in the Kermadec Trench 10,000 kilometers away than it does with its neighbors, just a few hundred kilometers away on the abyssal plain. This is not what we expected. We expected isolation to drive divergence, but instead we see convergence. The trenches, despite their extreme conditions, are somehow connected, or they share a common evolutionary history that has been preserved across vast distances. The reason for this, Drazen and Shank suspect, lies in the way organic matter reaches the hadal depths. The trenches act as traps, accumulating falling detritus from the surface waters above, and also capturing material that drifts down the continental slopes. This food is scarce, but it is consistent, and it has allowed a unique community of scavengers and deposit feeders to evolve, a community that is adapted to the specific conditions of the hadal zone, not to the surrounding abyssal plains. The amphipods in the trenches are not just the same species, they are part of the same ecological guild. occupying the same niche, and they have been doing so for millions of years. They are the Hadal specialists, and they are found nowhere else on Earth. But the Hades project has also revealed something even more surprising about the sediment in the trenches. Ronnie Glood, a microbial ecologist from the University of Southern Denmark, has been measuring the rates of carbon mineralization in the Hadal sediments, and he has found that they are remarkably high, far higher than the rates measured on the abyssal plains at similar depths. The microbes in the trench sediments are processing organic carbon at a rate that is out of proportion to the amount of food that is falling from above. This suggests that the trenches are not just passive traps, but active bioreactors, hotspots of microbial activity that are cycling carbon and nutrients at a pace that we did not anticipate. The implications are profound. If the trenches are processing carbon at these rates, then they are playing a much larger role in the global carbon cycle than we ever imagined, and they may be buffering the effects of climate change in ways we are only beginning to understand. But the Hadal zone is not just a place of sediment and microbes. It is also a place of geology, and the geology of the deepest trenches is intimately connected to the chemistry of life itself. In the 1980s and 1990s, a geologist named Patricia Fryer from the University of Hawaii made a discovery that would change our understanding of the deep sea. She was mapping the seafloor near the Mariana Trench, and she found something that looked like a volcano. But it was not a volcano in the traditional sense. It was a serpentinite mud volcano, a mound of soft serpentine rock that had been pushed up from the depths by the slow, churning motion of the Earth's mantle. These mud volcanoes are not erupting lava. They are oozing a thick, alkaline slurry of serpentine minerals and water, and they are found in a chain that runs along the edge of the trench, marking the boundary where one tectonic plate is diving beneath another. The process that creates these mud volcanoes is called serpentinization. When seawater seeps down through the fractures in the seafloor and comes into contact with peridotite, a rock that makes up the Earth's mantle, it triggers a chemical reaction. The seawater reacts with the minerals in the peridotite, producing hydrogen gas, methane, and a highly alkaline fluid. This is not a gentle reaction. It is a violent exothermic process that generates heat and transforms the rock into a soft, green, serpentine mineral. The resulting fluid is so alkaline that it can dissolve carbon dioxide, and it can sustain entire ecosystems of microbes that thrive on the hydrogen and methane, using them as a source of energy in a world without sunlight. Fryer and her colleagues have studied two of these mud volcanoes in detail, Conical Seamount and South Chamorro Seamount, both located in the Mariana Forearc, the region between the trench and the volcanic arc of the islands. These seamounts rise several kilometers above the seafloor, and they are covered in a thick layer of serpentine mud that is teeming with microbial life. The microbes are not the same as the ones found in the trenches, but they are equally fascinating because they are living in conditions that we once thought were incompatible with life. The pH of the fluids is around 12.5, which is as alkaline as bleach, and the temperatures are cold, around 4 degrees Celsius, yet the microbes are thriving, using the hydrogen and methane as their energy source. The connection between serpentinization and the origins of life is one of the most tantalizing ideas in modern science. The Lost City hydrothermal field, discovered in the mid-Atlantic in 2000, is a spectacular example of serpentinization at work. It is a field of towering carbonate chimneys, some reaching 60 meters in height, that emit fluids at temperatures of 40 to 90 degrees Celsius, and they are surrounded by dense communities of microbes that are living on the hydrogen and methane produced by the serpentinization reaction. The conditions at Lost City are remarkably similar to the conditions that we think existed on the early Earth, when the oceans were more acidic and the atmosphere was devoid of oxygen. Some scientists believe that life may have originated in exactly these kinds of environments, in the warm, alkaline fluids of a hydrothermal vent, where the chemical gradients between the vent fluids and the surrounding seawater could have driven the first metabolic reactions. The Hadal zone, then, is not just a graveyard of sinking detritus. It is a crucible of chemical and biological activity, a place where the fundamental processes of life are still being played out. And we are only just beginning to understand how these processes work. But as we learn more about the deep sea, we are also learning that it is under threat. And the threat comes not from the darkness or the pressure, but from our own activities on the surface. The Clarion-Clipperton Zone, a vast area of the Pacific Ocean that spans roughly 4.5 million square kilometers, lies between Hawaii and Mexico at depths of 4,000 to 5,500 meters. This is not a trench, but it is part of the deep sea, and it is covered in a carpet of polymetallic nodules, small potato-sized masses of manganese, iron, nickel, copper, and cobalt that have been accumulating on the seafloor for millions of years. These nodules are worth billions of dollars, and they are the target of a new gold rush, a rush to mine the deep sea for the metals that we need to build batteries, wind turbines, and solar panels. The International Seabed Authority, established under the United Nations Convention on the Law of the Sea, is the body that regulates mining in international waters, and by 2023, it had issued more than 30 exploration licenses for the Clarion-Clipperton Zone. These licenses allow companies to explore the area, to map the nodules, and to test their extraction techniques, but they do not yet allow commercial mining. The regulations for commercial mining are still being written, and there is a fierce debate about whether we should allow it at all. The concerns are well-founded. The Discal experiment, short for disturbance and recolonization, was carried out in 1989 in the Peru Basin, a small patch of the deep sea floor. A team of scientists dragged a plow across the seabed, mimicking the effect of a mining collector, and then they monitored the area to see how the benthic community recovered. They expected the tracks to be recolonized within a few years, but they were wrong. In 2015, 26 years after the disturbance, the tracks were still clearly visible, and the sediment was still not fully recovered. The plume of suspended sediment that would be generated by a mining operation is a particular concern. It could spread over hundreds of kilometers, smothering the Hadal communities that live in the trenches, which are downstream of the Clarion-Clipperton Zone. The sediment plume would not just affect the mining site, it would affect the entire ecosystem, from the abyssal plains to the deepest trenches. The Hadal communities that Jeff Drazen and Timothy Shank are studying are particularly vulnerable. They are already living in a food-limited environment, and they are not adapted to cope with pulses of sediment that would bury their feeding grounds or clog their filtering apparatus. The amphipods in the trenches are the most abundant animals in the deep sea, and they are the primary scavengers, cleaning up the carcasses that fall from above. If a sediment plume were to smother them, the entire Hadal food web would collapse, and the recovery would take centuries, if it ever happened at all. The deep-sea mining debate is not just about the environment. It is also about the future of our species. We face a global climate crisis, and we need the metals that are sitting on the seafloor to build the technologies that will help us transition away from fossil fuels. But we also face a biodiversity crisis, and we are on the verge of destroying an ecosystem that we have barely begun to understand. The Hadal Zone is a place of extraordinary life, and it is a place that we are about to alter forever. The water that fills the trenches is not static. It is part of a vast global circulation system, the thermohaline circulation, that connects the surface to the deep and the poles to the equator. Cold, dense water sinks in the polar regions, particularly in the North Atlantic and around Antarctica, and it flows along the bottom of the ocean, filling the basins and the trenches as it goes. This water is rich in oxygen because it was recently at the surface, and it is this oxygen that sustains the life in the Hadal Zone. The deep sea is not a stagnant pool. It is a living, breathing system, and the water that reaches the Challenger Deep, the deepest point in the Mariana Trench, is about 1,000 years old. This has important implications for the fate of pollutants. If we dump a chemical into the deep sea, it will not stay where we put it. It will be carried by the currents, which will distribute it throughout the ocean, and it will eventually reach the trenches, where it will be trapped in the sediment. The water that fills the trenches is old, and it is not replaced quickly, so pollutants can persist there for thousands of years. We are already seeing traces of human activity in the deepest parts of the ocean, in the form of plastic particles, synthetic fibers, and even the residue of nuclear bomb tests. The Hadal Zone is not a pristine wilderness. It is a repository of our history and our mistakes. But the deep sea is also a place of surprising beauty, and nowhere is that more evident than at the cold seeps, which are found along the continental margins, often near the trenches. The Japan Trench, which lies off the eastern coast of Japan, is one of the most seismically active places on Earth, and it is also a site of cold seeps, where methane and hydrogen sulfide seep out of the seafloor. These seeps are oases of life in the deep, and they are dominated by communities of tube worms and Calyptogena clams, which are not feeding on the water column, but on the bacteria that live inside their tissues. These bacteria are chemosynthetic, meaning they use the chemical energy of the methane and hydrogen sulfide to produce organic matter, just as plants use the energy of sunlight to produce sugar. The tube worms and clams are the visible signs of a hidden world, a world where life is not dependent on the sun, but on the chemistry of the earth itself. The cold seeps are not just fascinating for their biological communities, they are also fascinating for what they might tell us about the origins of life. The conditions at a cold seep are similar to the conditions at a hydrothermal vent, in that they involve a chemical gradient between the fluids seeping out of the seafloor and the surrounding seawater. And it is precisely these gradients that we think might have driven the first metabolic reactions on the early Earth. The idea is that life may have arisen in a place where the chemistry was just right, where the energy released by the mixing of different fluids could be captured and used to build complex molecules. The cold seeps and the hydrothermal vents are the modern-day equivalents of those ancient environments, and they are the closest we can come to observing the conditions that gave rise to life. But the cold seeps are also fragile, and they are vulnerable to the same disturbances that threaten the rest of the deep sea. The Japanese tsunami of 2011, which was caused by a massive earthquake on the Japan Trench, had a catastrophic effect on the communities living at the seeps. The turbulence of the earthquake and the tsunami resuspended sediment, which buried the tube worms and the clams, and the communities have been slow to recover. The event was a natural disaster, but it was also a reminder of how quickly the deep sea can be altered and how slowly it can recover. The turbidity currents that sweep through the trenches are another source of disturbance, and they are among the most powerful forces in the deep sea. These currents are underwater avalanches triggered by earthquakes, landslides, or the collapse of sediment slopes, and they can travel at speeds of 19 meters per second, far faster than any other current in the ocean. When a turbidity current passes through a trench, it deposits a thick layer of sediment called a turbidite, which can bury everything in its path. The communities living on the trench floor are not adapted to survive these events, and they are often killed instantly, buried under meters of sediment. But the turbidity currents are also a paradox, because they deliver pulses of organic material to the trench floor. The sediment that they carry is rich in organic matter, and when it settles, it provides a feast for the microbes and the detritivores that survive the initial disturbance. The trenches are thus characterized by a cycle of disturbance and recovery, where the communities are periodically wiped out and then rebuild over a period of years or decades. This cycle is a fundamental part of the Hadal ecosystem, and it is one of the reasons why the amphipods are so abundant, because they are adapted to take advantage of the pulses of food that follow a turbidity event. We are reminded, again and again, of the delicate balance that exists in the deep sea and of the forces that threaten it. The Hades Project has shown us that the trenches are not isolated, but are connected to the rest of the ocean through the circulation of water and the flow of organic matter. The serpentinite mud volcanoes have shown us that the chemistry of the Earth can create conditions that are conducive to life, and that the origins of life may be intimately tied to the geology of the deep sea. The deep sea mining industry is showing us that we are willing to destroy what we do not fully understand in the pursuit of short-term gain. And the thermohaline circulation is showing us that the deep sea is not a sink, but a system, and that the pollutants we release will eventually find their way to the deepest parts of the ocean. The cold seeps and the tube worms and the clams are a reminder of the tenacity of life, of the ability of organisms to find a way to survive in the most unlikely places. And the turbidity currents are a reminder of the power of the Earth, of the forces that shape the seafloor and the life that lives on it. We are learning, but we are also losing, and the question is whether we can find a way to balance our needs with the needs of the deep sea, whether we can learn to live in a way that does not destroy the very systems that sustain us. We are at a crossroads, and the choices we make in the next few decades will determine the fate of the Hadal Zone and of the entire ocean. The water that fills the trenches is 1,000 years old, and it carries the memory of everything we have done. But within that memory, there is a question that has haunted marine biologists since the first sounding lines touched the abyss. How deep can life go? And more specifically, how deep can a fish go? Because for all the strange creatures we have found in the Hadal Zone, for all the ghostly forms that drift through the perpetual dark, there is a line that no fish has ever crossed, and we think we know why. The answer lies in a molecule called trimethylamine N-oxide, or TMAO for short. It is a small organic compound that fish produce naturally, and it serves a remarkable purpose. In the deep ocean, pressure is a constant companion. At the surface, we feel 1 atmosphere of pressure, roughly 14 pounds on every square inch of our bodies. But at 8,000 meters, that pressure increases to nearly 800 atmospheres. The pressure would crush us instantly, but fish are not built like us. They are mostly water, and water does not compress. The problem is that their proteins and cell membranes are not water. They are delicate structures that would fold, twist, and tear apart under such enormous force. And that is where TMAO comes in. It accumulates in the tissues of deep sea fish and acts as a chemical stabilizer, preventing proteins from unfolding and membranes from losing their integrity. The deeper a fish lives, the more TMAO it produces. It is a perfect system, a beautiful adaptation, until it reaches a breaking point. Paul Yancey and his colleagues published a paper in 2014 that described this limit in extraordinary detail. They had been collecting fish from various depths across the Pacific, measuring their TMAO concentrations and plotting them against the depths where the fish were caught. The relationship was linear, almost perfectly so. As depth increased, TMAO increased. But the researchers noticed something alarming. If they extrapolated that line to its logical conclusion, there came a point where the TMAO concentration in a fish's tissues would become so high that it would no longer be compatible with life. The very chemical that protected the fish would become toxic. It would disrupt the osmotic balance of the cells, drawing water out of them and causing them to collapse. The fish would essentially dehydrate itself from the inside. That point, they calculated, was somewhere around 8,200 to 8,400 meters. And remarkably, the data from real fish confirmed it. No fish has ever been found below that depth. Not one. The deepest trenches extend to nearly 11,000 meters, but the fish simply stop. They cannot go further. It is not a physical barrier like a wall or a temperature gradient. It is a biochemical ceiling, a limit written into the very chemistry of life. But here is the strange thing. The amphipods that live in the deepest parts of the trenches are not subject to this same limit. Hirandelia gigas, the small shrimp-like crustacean that thrives in the Challenger Deep, has solved the pressure problem through a completely different biochemistry. Instead of relying on TMAO, it produces a different set of compatible solutes, compounds that stabilize its proteins without the same osmotic penalty. These molecules are less disruptive at high concentrations, allowing the amphipod to survive at pressures that would kill any fish. And so, at the bottom of the world, we find a world without fish. The vertebrates that dominate every other ocean habitat have been left behind. The amphipods rule alone. It is a humbling reminder that evolution does not always favor the most complex. Sometimes the simplest biochemistry wins. The implications of this are profound. When we think about the limits of life, we often think about temperature or radiation or the absence of water. But here, in the deepest ocean, the limit is a chemical one. It is a reminder that life is not just a matter of surviving pressure or cold. It is a matter of maintaining the delicate balance of chemistry that makes life possible. And that balance has a breaking point. We have found it. It sits at 8,400 meters, a line drawn in the water that no fish has ever crossed and no fish ever will. But the story of human exploration in the Hadal Zone is not just a story of limits. It is also a story of ambition. In November 2020, a small group of scientists and engineers gathered in the South China Sea, watching a submersible descend into the water. The vehicle was called Fenduje, which means striver in Chinese. It was a creation of the Institute of Deep Sea Science and Engineering in Sanya, a city on the southern coast of Hainan Island. And it was about to do something that only 3 other vehicles had ever done before. It was going to reach the bottom of the Challenger Deep. The first descent to full ocean depth was made by the Trieste in 1960, a Swiss-designed bathyscaphe crewed by Jacques Picard and Don Walsh. They spent 20 minutes on the bottom, saw a single flatfish, and returned to the surface. It was a remarkable achievement, but it was also a singular one. For the next 52 years, no one went back. Then, in 2012, James Cameron descended alone in the Deep Sea Challenger, a bright green vertical torpedo that he had helped design. He spent about 3 hours on the bottom, collected samples, and returned. And then in 2019, Victor Vescovo took the DSV limiting factor to the bottom, beginning a series of descents that would eventually visit every one of the world's deepest trenches. Fenduje was the 4th, and it was the first to carry Chinese scientists to the deepest point on Earth. The descent took several hours. The submersible, built to withstand pressures of more than 110 megapascals, carried 3 crew members. They spent time on the bottom, collecting sediment cores, capturing amphipods, and taking photographs of a world that had only been seen by a handful of people in all of human history. When they returned to the surface, they brought with them not just samples, but also a new sense of what was possible. The Chinese deep-sea program had arrived. The Fenduje program is part of a broader effort by the Institute of Deep-Sea Science and Engineering to understand the Hadal Zone. The institute, which was established in 2011, operates a fleet of research vessels and submersibles, including the Shenhai Yongshi, or Deep-Sea Warrior, which can dive to 4,500 meters. But Fenduje is the flagship, a vessel designed specifically for the deepest dives. Its development took years, and it required solving problems that had never been solved before. The hull had to be made of a special titanium alloy that could withstand the pressure without becoming brittle. The viewing ports had to be thick enough to resist the pressure, but clear enough to allow observation. The life support systems had to be reliable for descents that could last 12 hours or more. And the manipulator arms had to be precise enough to collect delicate samples from the seafloor without crushing them. The scientific data collected by Fenduje is still being analyzed, but early results have already changed our understanding of the Hadal Zone. The sediment cores taken from the Challenger Deep have revealed microbial communities that are more diverse than we expected. The amphipods captured have provided new insights into how life adapts to extreme pressure, and the photographs have shown us a seafloor that is not as barren as we once thought. There are tracks and trails in the sediment, evidence of creatures moving across the bottom. There are patches of white microbial mats, thriving on organic matter that has drifted down from the surface. And there are amphipods, thousands of them, swarming around the baited traps that the submersible deployed. The Chinese program is not alone in its efforts. Scientists from Japan, the United States, the United Kingdom, and other nations are also exploring the Hadal Zone. But the pace of discovery is accelerating, and each new descent brings us closer to understanding the deepest places on our planet. Yet for all that we have learned, there is one aspect of Hadal life that remains almost entirely unknown: reproduction. No researcher has ever observed mating or reproductive behavior in Hadal organisms in their natural environment. Not once. We have no idea how these creatures find each other in the dark, how they court, how they mate, or how they care for their young. We have no idea how often they reproduce, or how many offspring they produce, or how those offspring survive in the immense pressure and perpetual darkness of the deep. What we do know comes from 2 sources. The first is captured specimens. When researchers bring amphipods or other Hadal organisms to the surface, they sometimes find that the females are carrying eggs. The eggs are small, usually less than a millimeter in diameter, and they are often attached to the female's body in a brood pouch. The presence of these eggs tells us that the females must mate at some point, but it does not tell us how. The second source of information is closely related shallow water relatives. Many Hadal species have cousins that live in the deep sea, but not at Hadal depths, or even in shallow coastal waters. By studying these relatives, we can make educated guesses about how Hadal organisms might reproduce. But educated guesses are not the same as knowledge. The challenge is that Hadal organisms are incredibly difficult to keep alive. When they are brought to the surface, they experience a rapid decrease in pressure. Their bodies, adapted to the crushing weight of the ocean above, expand. Their proteins, stabilized by high concentrations of TMAO or other compatible solutes, begin to unfold. Their cell membranes, which were fluid at high pressure, become rigid and brittle. Most die within minutes of reaching the surface. Some die before they even reach the deck of the ship. And so we are left with a paradox. We can collect these creatures, but we cannot observe them. We can study their bodies, but not their lives. In recent years, researchers have developed a solution of sorts. Isobaric samplers are pressure vessels designed to keep specimens at their native pressure during retrieval. They are essentially underwater containers that can be opened at depth, allowing a specimen to swim or be swept inside and then sealed before the sampler is brought to the surface. The pressure inside the sampler is maintained at the same level as the depth where the specimen was collected. In theory, this allows the specimen to survive the journey to the surface. In practice, it has been only partially successful. Some specimens have survived. They have been observed alive in the laboratory, swimming in pressure vessels that replicate their native environment. But these successes are rare. The samplers are difficult to deploy, and they can only hold a limited volume of water. And even when a specimen survives, it is often stressed, and it rarely reproduces in captivity. So we are left with inference. We know that amphipods, which are the most common hadal organisms, have a reproductive cycle that involves molting. The female sheds her exoskeleton, and shortly after molting, she is receptive to mating. The male deposits sperm, and the female stores it until she is ready to fertilize her eggs. The eggs are then carried in the brood pouch until they hatch. The young amphipods emerge as miniature versions of their parents, and they grow by molting repeatedly throughout their lives. We know this because we have observed it in shallow water amphipods, which are closely related to their hadal cousins, but we do not know if hadal amphipods follow the same pattern. We do not know if they reproduce seasonally or continuously, or if they have a breeding season at all. We do not know if they migrate to find mates, or if they simply encounter each other by chance in the vast darkness. The unanswered questions in hadal biology are numerous. We still do not know whether any megafauna, any large animal, lives below 8,400 meters other than amphipods and holothurians, the sea cucumbers. We have found amphipods at the very bottom of the Challenger Deep, and we have found holothurians in the sediment, but we have not found any fish, any squid, any octopus, or any other large animal. The absence of fish is well explained by the TMAO ceiling, but the absence of other megafauna is more puzzling. Perhaps the food supply is too limited. Perhaps the pressure is too great for any animal larger than a few centimeters. Or perhaps we simply have not looked hard enough. The hadal zone is vast, and we have only sampled a tiny fraction of it. We also do not know the maximum depth at which any multicellular animal has been found. The current record holder is an amphipod, found at a depth of 10,900 meters in the Mariana Trench, but there may be other animals living even deeper. The trenches reach nearly 11,000 meters, and we have not yet explored the deepest parts of all of them. There are over 30 trenches in the world's oceans, and only a handful have been sampled. Some of the deepest trenches, such as the Philippine Trench and the Kermadec Trench, have been visited only a few times. The possibility of undiscovered life in these unexplored depths is real. The microbial communities in different trenches present another mystery. Are they isolated from each other, or are they connected? The trenches are separated by thousands of kilometers, and the deep currents that flow through them are slow and weak. It seems unlikely that microbes could travel from one trench to another, but recent genetic studies have found that some microbial species are present in multiple trenches. This suggests that there is some connection, perhaps through the global deep ocean circulation, or perhaps through the bodies of animals that migrate between trenches. The question is not yet settled. And then there is the question of age. How old are the oldest hadal organisms living today? The hadal zone is a place of extreme scarcity. Very little food reaches the bottom of the trenches. The organic matter that falls from the surface is mostly consumed by the time it reaches the deep sea, and what remains is a thin, sporadic rain of particles. This means that hadal organisms have very little energy to fuel their growth and reproduction. They may live for a very long time. Some deep-sea organisms, such as the tube worms that live at hydrothermal vents, are known to live for centuries. It is possible that hadal amphipods, with their slow metabolism and limited food supply, also live for decades or even centuries, but we have no way to measure this. We have no way to age a hadal amphipod. We can only guess. The dispersal mechanisms that allow similar species to appear in trenches separated by thousands of kilometers are also unknown. We find the same species of amphipod in the Mariana Trench, the Japan Trench, and the Peru-Chile Trench. How did they get there? The trenches are isolated from each other by vast expanses of abyssal plain, which is much shallower than the hadal zone. The amphipods cannot survive at those shallower depths. They cannot swim across the abyssal plains, and they cannot be carried by currents that do not flow through the trenches. So how did they spread? Perhaps they were once connected, and the trenches were part of a continuous hadal environment that has since been separated by tectonic activity. Or perhaps they are carried by the bathypelagic organisms that migrate between the surface and the deep sea. Or perhaps they are not truly the same species, but rather similar species that have evolved separately in each trench. The question remains open. When we extract a core of hadal sediment, a cylinder of mud 10 to 50 centimeters long, brought up from 11,000 meters, we find that the sediment is not uniform. The top few millimeters are inhabited by a community of meiofauna, organisms that range in size from 45 micrometers to 1 millimeter. These are the nematodes, the foraminifera, the copepods, and the chinorhynchs. They are tiny, but they are abundant. In a single square centimeter of hadal sediment, there can be thousands of these organisms. They feed on the organic matter that settles on the seafloor, and they are the foundation of the hadal food web. Below the top few millimeters, the sediment transitions to a zone dominated by bacteria and archaea. These microorganisms are found throughout the sediment column, but they become increasingly abundant as the meiofauna disappear. The bacteria and archaea are responsible for the decomposition of organic matter, and they play a crucial role in the cycling of nutrients. They are also the only organisms that can survive in the deepest layers of the sediment, where the pressure is highest and the organic matter is most degraded. The rate at which organic material is buried versus consumed is a key parameter in understanding the hadal carbon cycle. At the surface of the sediment, organic matter is consumed rapidly by the meiofauna and the bacteria. But as the sediment is buried, the organic matter becomes less accessible, and the rate of consumption decreases. Some organic matter is buried deep enough that it is effectively removed from the biosphere, sequestered in the sediment for millions of years. This is one of the reasons that hadal sediments are so important for understanding the global carbon cycle. They are a sink for carbon, a place where organic matter is stored for geological timescales. Stable isotope analysis of sediment cores tells us about past productivity at the surface. The ratio of carbon isotopes in the sediment reflects the source of the organic matter. If the organic matter came from phytoplankton, it will have a different isotope signature than if it came from seafloor microbes. By analyzing the isotope ratios in a sediment core, we can reconstruct the history of productivity in the surface ocean over millions of years. We can see periods of high productivity when the ocean was rich in nutrients and the phytoplankton bloomed, and periods of low productivity when the ocean was barren. The hadal sediment is a geological archive of ocean history, and it goes back millions of years. If we could sample every square meter of hadal sediment, sequence every organism's genome, and monitor every community for a decade, what would we learn? We would learn the true diversity of life under pressure. We would learn how many species exist in the hadal zone and how they are related to each other. We would learn whether life can originate chemosynthetically without sunlight. The hadal zone is dark, but it may contain environments where chemical reactions can provide energy for life. We would learn how contamination pathways work in detail, how pollutants from the surface are transported to the deep ocean, and how they affect the organisms that live there. We would learn the exact role of trenches in long-term carbon sequestration and how much of the carbon that we emit into the atmosphere ends up in the deep ocean. And perhaps most importantly, we would learn what the limits of life truly are on this planet and by extension on others. The hadal zone is the last frontier on Earth, and it holds the key to understanding the potential for life elsewhere in the universe. If life can survive at 11,000 meters, under pressures that would crush a submarine, in perpetual darkness with almost no food, then maybe life can survive anywhere. And that is a question that extends far beyond the bounds of our own planet. But we are not there yet. We are still at the beginning, still taking our first steps into the abyss, still descending into the dark with our lights and our cameras and our samplers, trying to understand a world that is almost entirely unknown to us. And perhaps that is the most remarkable thing of all, that after all these years, after all the descents and all the samples and all the analysis, we still know so little. We are still standing at the edge of the abyss, looking down, wondering what we will find when we finally go all the way down. The ocean is not a silent place. It is filled with a vast and continuous chorus of sounds, from the crackle of snapping shrimp to the low moans of whales, from the rumble of underwater earthquakes to the distant thunder of storms breaking on the surface far above. But there is a particular depth, a specific layer of the ocean, where sound behaves in a way that is almost magical. This is the SOFAR channel, the sound fixing and ranging channel, and it exists roughly between 700 and 1,000 meters below the surface. In this zone, a delicate balance between temperature and pressure creates a condition where sound waves are bent and trapped, traveling with an astonishingly minimal loss of energy. The result is an underwater highway for sound, a whispering gallery that can carry a single noise across an entire ocean basin. We can imagine the physics of it, the way the speed of sound in water is not constant but varies with temperature and pressure. Near the surface, the water is warmer and sound travels faster. Deeper down, the water is colder and sound travels more slowly. But at a certain depth, the pressure begins to dominate, and the speed of sound starts to increase again. The point where the speed is at its minimum creates a lens, a natural waveguide that keeps sound from spreading upward or downward. Instead, it stays trapped in this narrow horizontal band, and it can travel for thousands of kilometers without fading away. This is a phenomenon that we have only recently learned to appreciate, and it has shaped our understanding of the ocean in ways we never anticipated. The discovery of this channel came during the middle of the 20th century, when the world was divided by the Cold War. Scientists working for the military were trying to understand how to detect submarines, and they stumbled upon this remarkable acoustic property of the deep ocean. They realized that if you placed a small charge of explosives in the SOFAR channel and detonated it, the sound would travel for thousands of kilometers, and it could be heard at listening stations on distant shores. This became a tool for locating downed pilots, a way to send a signal that could be detected over vast distances. The system was called SOFAR, and it was a way of using the ocean itself as a medium for communication and navigation. But the military use was just the beginning of our understanding, and the channel would soon reveal secrets that had nothing to do with human conflict. We began to notice that the ocean was already using this channel long before we ever knew it existed. The great whales, the blue whales, and the fin whales produce calls that are low in frequency and incredibly powerful. These calls are perfectly suited to the SOFAR channel, and they can travel across entire ocean basins, from one side of the Atlantic to the other, without losing their integrity. We think of whales as solitary wanderers, but they are actually connected by this invisible network of sound. A blue whale in the Pacific can call to another blue whale 1,000 kilometers away, and the message will arrive intact, a deep and resonant song that travels through the dark water at the speed of sound. This is not a coincidence. The whales have evolved to use this channel, and they have been communicating through it for millions of years, long before we ever had the technology to listen. But below the SOFAR channel, in the Hadal depths, the acoustics change. The pressure at these depths is immense, and the water becomes denser. This density affects the speed of sound, causing it to increase again. The sound waves that enter the Hadal zone are no longer trapped in a narrow channel. They spread out, they lose energy more quickly, and they behave differently than they do in the shallower waters above. This has profound implications for how we communicate with the vehicles we send down to the deepest trenches. A remotely operated vehicle, or an autonomous lander, cannot rely on radio waves to transmit data back to the surface. Radio waves do not travel through water, at least not in the way we need them to. Instead, we use acoustic signals, sound waves, to send commands and receive data. But in the Hadal zone, these acoustic signals are distorted by the pressure and the density, and they cannot travel as far or as clearly as they do in the SOFAR channel. This means that when we send a vehicle down to the deepest point of the Mariana Trench, we are essentially sending it into a region of acoustic isolation. The vehicle must operate on its own, following pre-programmed instructions, because the communication link with the surface is slow and unreliable. The acoustic signals travel at the speed of sound, which is about 1,500 meters per second, and there is a time delay and a loss of clarity that makes real-time control nearly impossible. We have to trust our machines to do their work autonomously, and we have to wait for them to return to the surface to learn what they have found. This is a kind of faith, a willingness to relinquish control and allow the technology to operate in an environment where we cannot see it and cannot hear it clearly. The deep ocean is not silent, even at these depths. The NOAA hydrophone network, a system of underwater microphones deployed around the world, has captured a vast array of sounds from the deep ocean. These sounds include the low rumble of earthquakes, the crackling of ice, the calls of whales, and the constant background noise of the ocean itself. But in 1997, the network captured a sound that was so strange and so powerful that it became the subject of intense speculation. This sound was called the bloop, and it was picked up by hydrophones thousands of kilometers apart. It was a low-frequency sound, incredibly powerful, and it did not match any known source. The speculation was immediate and wild. Some people suggested it was the sound of a giant creature, a creature so large that it had never been seen, lurking in the depths of the ocean. The idea captured the imagination, and the bloop became a legend, a mystery that seemed to defy explanation. But the truth, as it often is, was more mundane and more fascinating. The bloop was eventually identified as the sound of a large iceberg fracturing. When an iceberg breaks, it can produce a sound that is incredibly powerful and that travels for thousands of kilometers through the ocean. The sound of the bloop was consistent with the sound of ice, and as our understanding of the ocean improved, we realized that the great ice sheets of Antarctica were the source. The mystery was solved, but the solution was no less remarkable. The idea that a piece of ice breaking apart in the remote southern ocean could produce a sound so powerful that it was heard across an entire ocean basin is a testament to the strange and wonderful acoustics of the deep. The ocean is a medium of connection, and it carries the sounds of its own processes, the cracking of ice and the rumbling of the earth, across distances that seem impossible to our terrestrial minds. As we think about the acoustics of the deep ocean, we are also confronted with the limits of our knowledge about the ocean floor itself. We have mapped the surface of Mars and the surface of the Moon in greater detail than we have mapped the bottom of our own oceans. As of the early 2020s, only approximately 24% of the global ocean floor had been mapped with high resolution, which means a resolution better than 100 meters per pixel. This is a shocking statistic, and it speaks to how little we truly know about the basic geography of our own planet. The ocean floor is vast and remote, and it is difficult to reach. We have sent more people to the Moon than we have sent to the deepest parts of the ocean, and we have more detailed maps of the surface of Venus than we have of the seafloor. This is why the Seabed 2030 project was launched. In 2017, the Nippon Foundation and the General Bathymetric Chart of the Oceans, which is known as GEBCO, came together with an ambitious goal. They wanted to map the entire ocean floor by the year 2030. This is a monumental undertaking, and it requires the cooperation of nations, institutions, and individuals around the world. The project is not just about creating a map for the sake of having a map. It is about understanding the fundamental structure of our planet, the way the ocean floor is shaped by tectonic processes, the way it provides habitat for life, and the way it influences the circulation of the ocean and the climate of the Earth. The ocean floor is not a flat and featureless plain. It is a dynamic landscape with mountains and valleys, volcanic ridges and deep trenches, and it is teeming with life in ways that we are only beginning to understand. The challenge of mapping the ocean floor is the challenge of reaching it. The methods we use to map the ocean floor are very different from the methods we use to map the surface of a planet. We cannot send a satellite to take photographs of the seafloor because water is opaque to most forms of electromagnetic radiation. The light from the sun can only penetrate a few hundred meters into the ocean, and below that, everything is dark. To map the ocean floor, we have to use sound. A ship at the surface sends out a sound wave and it bounces off the seafloor and returns to the ship. By measuring the time it takes for the sound to return, we can calculate the depth of the ocean at that point. This is called acoustic mapping, or sonar, and it is the most accurate way we have of measuring the depth of the ocean. But this method is slow and expensive. A ship can only map a relatively narrow swath of the ocean floor as it travels, and it takes a long time to cover the entire ocean. This is why we have also used satellite altimetry to get a rough estimate of the seafloor topography. A satellite in orbit around the Earth can measure the height of the sea surface with remarkable precision. The sea surface is not flat. It bulges slightly over underwater mountains because the gravitational pull of the mountain's mass pulls water toward it. It dips slightly over underwater troughs because the gravitational pull is weaker. By measuring these variations in the sea surface height, we can infer the presence of features on the ocean floor, but the resolution is much lower than what we can achieve with direct acoustic mapping from a ship. The satellite data gives us a general picture, a rough outline, but it cannot show us the details. The difference between these two methods is enormous. The satellite data is like looking at a map of a continent from space, where you can see the major mountain ranges and the outlines of the coastlines, but you cannot see the individual valleys and peaks. The acoustic data from a ship is like walking through that continent on foot, seeing every detail of the terrain. The Seabed 2030 project aims to fill in the gaps, to use ships and other platforms to map the ocean floor with high resolution, and to compile the data into a single, comprehensive map. But the ocean is vast, and the time is short. The project is an ambitious one, and it will require the contributions of many people and many nations to succeed. Even if we do not achieve the goal by 2030, the project has already revealed how much we have to learn, and how much of the ocean floor remains unknown. As we contemplate the unknown geography of the deep, we are also confronted with the ways in which the deep ocean is changing. The ocean is warming, and the effects of climate change are reaching even the deepest parts of the ocean. The thermohaline circulation, the great conveyor belt of the ocean that carries cold, oxygen-rich water from the poles to the deep sea, is a vital system. This circulation is driven by differences in temperature and salinity, and it delivers oxygen to the deepest trenches, allowing life to exist in the Hadal zone. But as the ocean warms, the thermohaline circulation may weaken. The surface waters are becoming warmer and less dense, and they are becoming more stratified, which means there is less mixing between the surface and the deep. This could reduce the supply of oxygen to the deep ocean, and it could have profound consequences for the life that depends on it. We are already seeing evidence of deoxygenation in some intermediate depth zones. The oxygen minimum zones, the layers of the ocean where oxygen levels are naturally low, are expanding and intensifying. These zones are found at depths between a few hundred and 1,000 meters, and they are already stressful environments for marine life. As the ocean warms, these zones may expand, and the oxygen may be depleted even further. The Hadal zone itself is buffered by its depth from direct warming. The bottom of the Mariana Trench, at almost 11,000 meters, is not directly affected by the warming of the surface waters. The temperature at the bottom of the trench is just above freezing, and it is stable. But the indirect effects of climate change are reaching the Hadal zone, and they are changing it in ways that we are only beginning to understand. The surface productivity is changing. The phytoplankton, the microscopic plants that form the base of the marine food web, are becoming more or less abundant in different parts of the ocean, and this affects the amount of organic matter that sinks to the deep sea. The marine snow, the constant rain of organic particles that falls from the sunlit surface waters to the dark depths, is composed of these phytoplankton, along with the fecal pellets of zooplankton and the remains of other organisms. This marine snow is the primary food source for life in the deep ocean, and if its composition changes, the life that depends on it will change as well. The altered marine snow may be more or less nutritious, and it may carry different amounts of organic carbon, and this will affect the entire ecosystem of the Hadal zone. The reduced circulation of the ocean will also have an effect. If the thermohaline circulation weakens, the deep ocean will become more stagnant, and the oxygen levels will drop. This will make the Hadal zone more stressful for the organisms that live there, and it may reduce the amount of life that can survive. The Hadal zone is home to organisms that have been stable for thousands of years, adapted to a constant environment of extreme pressure, near freezing temperatures, and complete darkness. These organisms are not adapted to change, and they may be vulnerable to the indirect effects of a warming climate. The changes will be slow, measured in centuries rather than years, but they will be profound. The deep ocean is not immune to the changes we are causing on the surface, and the consequences will be felt in the most remote and inaccessible places on Earth. We think about the life in the Hadal zone, and we wonder about the light. In the ocean above 1,000 meters, bioluminescence is the primary source of light. 76% of deep-sea species can produce their own light, using a chemical reaction that emits a soft glow in the darkness. This light is used for communication, for camouflage, for attracting prey, and for confusing predators. It is a world of living light, a constant display of biological fireworks in the dark water. But below 1,000 meters, in the abyssal and Hadal zones, the organisms captured by landers show far fewer bioluminescent structures. The deep-sea creatures that we have brought to the surface from the Hadal zone are not as luminous as their shallower relatives. This is a puzzle, and we do not fully understand why. Whether bioluminescence exists in the Hadal zone is not clearly known. The cameras used in lander deployments would detect it if it were present, but most observations occur in bright artificial light that would overwhelm any faint bioluminescent signals. When we send a lander down to the bottom of a trench, we turn on the lights, and we see the creatures that are there, but we cannot see whether they are producing their own light because the artificial light is so much brighter. The possibility remains that Hadal organisms are bioluminescent, but that we simply have not seen it. It is also possible that they have evolved to communicate in other ways. In the absence of light, in the complete darkness of the Hadal zone, organisms may rely on chemical signals or mechanical signals, sensing the chemical traces of other organisms in the water, or feeling the vibrations of their movements. The deep sea is a world of the senses, and the senses that are used in the Hadal zone may be very different from those we are familiar with. The last decade of Hadal science has transformed our understanding of the deep ocean. In 2010, The standard view of the Hadal Zone was that it was sparsely populated and relatively uniform. It was thought of as a biological desert, a place of little life and little interest. But by 2024, that view had been completely transformed. Hadal trenches are now understood to be biologically diverse, chemically complex, ecologically important systems that are connected to the surface through multiple pathways. The thermohaline circulation brings oxygen and nutrients to the deep. The turbidity currents carry sediment and organic matter down the slopes of the trenches. The whale falls provide a sudden and abundant source of food for scavengers, and the slow rain of marine snow provides a constant, if meager, supply of organic carbon. The Hadal Zone is not isolated. It is a part of the global ocean system, and it is connected to the surface in ways that we are only beginning to appreciate. The number of described Hadal species has increased significantly. We have discovered new species of fish, new species of amphipods, new species of microbes, and new species of many other organisms. The Hadal Zone is not a desert. It is a unique habitat, home to a distinct community of organisms that are found nowhere else on Earth. The contamination story has fundamentally changed the narrative. We have found plastic fibers in the guts of Hadal amphipods, taken from the deepest parts of the ocean. We have found persistent organic pollutants in the tissues of Hadal organisms, chemicals that were banned decades ago, but that have found their way to the deepest trenches. The idea that the Hadal Zone is pristine and untouched by human activity is a myth. The contaminants that we have released into the environment are reaching the deepest parts of the ocean, and they are accumulating in the food web. The depth record for fish has been broken multiple times. We have seen fish at depths that were previously thought impossible. The snailfish, the deepest living fish we have found, has been observed at depths of more than 8,000 meters. This is a remarkable discovery, and it challenges our understanding of what is possible for vertebrate life. The pressure at these depths is immense, and the adaptations that allow these fish to survive are extraordinary. And we have come to realize that these systems are already being affected by human activity, despite being as remote as any place on Earth. The deep ocean is not a refuge from the consequences of our actions. It is a part of the system that we are changing, and it is feeling the effects. We are learning that the Hadal Zone is a dynamic and active environment, not a static and unchanging one. The trenches are places of geological activity, where the tectonic plates meet and grind against each other. They are places of earthquakes and landslides, of turbidity currents and sediment flows. They are places where the chemistry of the ocean is altered, where the carbon that falls from the surface is processed and buried. The Hadal Zone plays a role in the global carbon cycle, and it is important for the regulation of the Earth's climate. The deep ocean is not a distant and irrelevant place. It is a part of the planet that we live on, and it is connected to everything that we do. As we continue to study the Hadal Zone, we are discovering that it is far more complex, far more interesting, and far more important than we ever imagined. And as we learn more, we realize that there is still so much we do not know, and the mysteries of the deep. In the summer of 2023, a camera system descended into the Izu Ogasawara Trench, south of Japan, and recorded a single snailfish, Pseudoliparis bellia avi, moving across the sediment at 8,336 meters. The footage lasted seconds. It confirmed the species existed at that depth, and then the camera returned to the surface. The fish, presumably, remains. It is doing what it does in the darkness, feeding, avoiding, moving, in the 99.99% of its existence during which no instrument is watching. We have a name for it. We have a depth for it. We do not have a biography. No one knows how it finds its food, how it mates, how it navigates the total absence of light, or how long it lives. The species was described from that single encounter, and that description is the entirety of our knowledge. The Hadal Zone will outlast the institutions that study it. The pressure at those depths will not diminish. The trenches are geological facts, carved into the planet's crust over millions of years, and they will continue to exist long after every research program, every funding cycle, and every expedition log has been archived and forgotten. The sediment will continue to accumulate. The cold will remain constant. The trenches do not acknowledge the science conducted within them. That is not a statement of defeat. It is a statement of scale. The incompleteness of our knowledge is not a failure. It is a measurement. The Hadal Zone is vast, and our instruments are few. Every species described from a single specimen is a door opened, not a room explored. Every depth record is a boundary crossed, not a destination reached. The science is unfinished because it has only recently begun. The first manned descent into the Challenger Deep happened within living memory of some of the people who watched it on television. The snailfish was filmed only last year. This is not the end of a story. It is the first page. The trench is there. The animals are there. The pressure is constant. The camera will descend again, and when it does, it will encounter something we did not expect, and we will name it, and we will understand slightly more than we did before. And the trench will remain, indifferent to our understanding, vast and constant, and the science will continue.